minutes. I can see Madam joining.
Yes.
Yeah, Madam is joining. Good morning, Madam.
Good morning, Nithya. Good morning.
Good morning. Good morning.
Good morning, Dr.
Good morning,
Nilima.
Raju ji. Good morning, sir.
Good morning. Mahesh, please get started. Yeah.
Yes.
9:30. Yeah.
Has Madam Sangeeta Rawat joined?
Madam, she's not today, unable to join today, but she has sent us a recorded message, Madam. We'll show it.
Wonderful. Very good.
Mahesh, get started, please.
Yes, ma'am. Good morning, all. I'm Dr. Mahesh Belekar, Associate Professor in the Department of Clinical Pharmacology at GS Medical College and KEMH Hospital, Mumbai. On behalf of our department, I welcome all the chairpersons, speakers, and delegates for today's advanced level training program entitled The Art and Science of Benefit Risk Analysis. Our Dean Madam is unable to be with us, has recorded a message for all of us, which we will play now. I request our IT team, please proceed.
Good morning to everybody, and at the outset I would like to warmly welcome all of you to this advanced level of workshop on pharmacovigilance, which is titled as Art and Science of Benefit Risk Analysis. It is organized by Clinical Pharmacology Department under the able leadership of Dr. Nithya Gupta, and it's under the Pharmacovigilance Program of India. Also, I would warmly welcome the speakers, the chairpersons, and the audience who have turned into a live YouTube, who have joined for this program. So this 4-hour program is really going to be beneficial to all of you in knowing what is the risk involved with anti-epileptic drugs, antibiotics, and many others. The this vigilance, I think, started in KEM way back in 1980, and I was in close association when I joined neurology residency. After that, I remember referring patients with anti-seizure medications and side effects. Especially, I remember phenytoin causing, we used to think that this phenytoin has caused ataxia, but it was actually the phenytoin and phenobarb long-term causing cerebellar atrophy. And then these patients would confirm their toxicity in some of the blood levels, which this department was doing since so many years. It was very remarkable, till now it is still helping us in doing the serum levels of this anti-epileptic drugs. And they would then get registered in the national database and will be reported to higher authorities. So definitely this pharmacovigilance, I mean, whenever we are giving a drug, the side effects are something which we must know. I have followed one principle that whenever patient comes and tells you that this drug is causing a problem, always believe the patient. This may not be a known side effect which you know it from your science, but whenever if patient tells and you check your our literature, there will be some case report of this reported somewhere. So this has really helped that if the patient comes and tells you that this is what I have got, which is not a common side effect, but it may be an uncommon side effect. So never forget and never unbelieve the patient. Always try to believe patients and check whether these are the adverse effect which is caused by that drug. And if it is not and patient is very sure, try and report it to departments like this where they can definitely establish and report as a new adverse effect of this drug. And I'm sure this is a half-day program, but very carefully planned by her team. And this will definitely enhance our knowledge on pharmacovigilance, basically the side effects of the drugs. And it will tell us new dimensions, which now we have also started, I think, here at KEM, that pharmacogenetics, because some of the patients are genetically more prone to develop side effects. So now we also are undertaking studies on pharmacogenetics, so that we know this. These are the patients who have problem with because of the genetic aspect having side effects, and these then can never be given those certain drugs because they have a genetic abnormality. So if we know this, definitely it helps us in choosing a better drug with less side effects. So once again, I welcome all of you, and I'm sure this 4 hour, next 4 hour are going to be very, very fruitful, and you will definitely go from new knowledge and new concepts about this pharmacovigilance. And I wish all the best for this whole program. Thank you.
We are thankful to our Dean Madam. Thank you, Madam, for her support and guidance for today's program. Now I welcome and introduce our first speaker, Dr. Raju Singh Ravenchi. Sir is presently Secretary Scientific Director at Indian Pharmacopoeia Commission. Sir has more than 30 years of experience in drug development and was previously associated with Dr. Reddy's Laboratories as a Senior Vice President and Head. He was former Director at Ranbaxy Research Laboratories and was Scientist at National Institute of Immunology. He has also served as a Research Associate of Formulation Development at Dabur Research Foundation. I request Sir to proceed with his talk on Pharmacovigilance Program of India, that is PVPI, a perspective from Indian Pharmacopoeia Commission. I request Sir to proceed.
Thank you. Thanks a lot, Dr. Mahesh. And good morning to all the participants. Respected Dr. Nilima, Dr. Nithya, and the organizing team. Thanks a lot for giving this opportunity to come and engage with this group of professionals involved in pharmacovigilance activities. It's certainly a privilege for me to put some of the information and perspective from Pharmacovigilance Program of India, which is being run by IPC for the government. So I'll just take a second to share I have a few slides, and I'll take help of those slides to do the presentation and maybe some discussion after that. Is my screen visible?
Yes, sir.
Yes, sir. Please go ahead. Yeah.
Yeah. Great. Thank you. So couple of minutes on IPC, because this won't be justice for me if I don't talk about IPC, the organization which I head and the organization which is managing the Pharmacovigilance Program of India. So IPC is an autonomous body under Ministry of Health and Family Welfare, Government of India. And it came into existence on 1st January 2009. And it is organized like many other organizations, where we have the highest body called general body, and then the governing body and scientific body. And then we have different groups managing different activities at IPC. These are the 4 mandates of Indian Pharmacopoeia Commission. The Pharmacovigilance Program of India is one of the more important activities which we manage for India. And apart from Pharmacovigilance Program, the mandate is also to bring out the Indian Pharmacopoeia, the book of pharmaceutical standards for this country. We put in all the monographs for drugs which are being sold in this country with their standards, their method of analysis, quality checks, and other related parameters. We also develop and make available reference standards for all the stakeholders, mainly the pharma manufacturers, for analyzing their products which they are manufacturing. These reference standards are used against which you check your quality. And a very important program is also publishing National Formulary of India, which contains many clinical monographs, covers all the drugs which are part of the NLEM, all the drugs which are part of Pharmacopoeia, and other drugs which are being used in national health programs. So very important and very concise information in the form of National Formulary of India. So today, this morning, I would focus on the Pharmacovigilance Program of India. And so when it comes to Pharmacovigilance Program of India, this started in a structured way around 2010 at AIIMS. And then from there, it was transferred to IPC in April 2011. And since it came to IPC, it's almost 11 years now. Sorry. And in last 11 years, we have actually done a remarkable job as a team. And with the help of experts, a lot of you have contributed to the growth of Pharmacovigilance Program of India in this country. Currently, there are 4 major sources of the adverse events which we are collecting in the program. The first one is the adverse event monitoring centers which we have created throughout the country. I'll come to that in subsequent slides. Then a major source is also the marketing authorization holders who are selling drugs in this country after approval from CDSCO. And as a rule, they are expected to report all the adverse events coming out of their drugs, which they are selling, till the drug is in the category of new drug. Then we get a lot of adverse events reporting from national health programs like NACO, managing the AIDS program, the National Tuberculosis Mission, the Leprosy Mission, the Vector-borne diseases mission, and so on. And then there is also a chunk of the patients or their family members directly reporting. And this area currently in this country is very small. And maybe because of the awareness which the patients have, they still are not participating to the extent they should. But certainly we are doing some strategies to bring them directly into the value chain. So this bar diagram shows how we grew from 58 adverse event monitoring centers in 2011 to currently we have actually operationalized 505 of them, a huge increase in last 10-11 years. And what I am told that this 505 is now 534, and we expect that we should be touching around 600 centers by end of this financial year. So that's a huge growth and spread of the network of AMCs in the country, which is helping us increase the awareness among the stakeholders and also collecting a lot of adverse event data and sending it to IPC for analysis. This map shows the distribution of the AMCs, and you would appreciate that this is spread across the country. Almost every state of the country has multiple AMCs, barring some of these which we are currently putting efforts to start at least one center in these small states also. But what is important to note here, that right from Jammu and Kashmir to Kanyakumari or Kerala, and from Gujarat to Mizoram and Manipur, we have established these centers. This is the trend of the number of reportings coming every year. And last calendar year, which is 2021, we closed around 70,000. So if you see from 2016 onwards, the trend is almost plateauing around 70,000, little more, little less. And this need to change. And we are doing a lot of efforts and strategies to make it increase significantly in the coming year. And we have put in a target of 100,000, which is an important milestone for PVPI program if we hit 100,000 reportings in 1 year for the first time. And that would certainly motivate and boost the morale of all the stakeholders working for PVPI. But the good part is, from where we started with around 15,000 16,000 reportings. Today, we are around 70,000. So here also, along with the number of centers increasing, the number of reportings have also increased. This is just a distribution of the AMCs which have been reporting the ICSRs or adverse events. And certainly area of concern, there are very few centers which are actually reporting to the extent we would have expected. Even if I take a cutoff of say 500 reportings in a year, which is just 30-40 reportings per month, we see that around 50 centers out of 505 fall into that category, not even 50. And a major chunk of adverse event reporting centers, they are either not reporting or they are reporting less than 100 in a year. That means less than 10 reportings per year, per month, which is certainly a area to be looked into, and certainly a concern for us from the regulator and government side. And this area is something which we are very vehemently discussing among all the stakeholders. Wherever I speak, I request stakeholders like all of you to help us shift this whole bar towards right, so that this area increases. This is certainly a concern. But certainly a positive indicator is what we did was we took last 5 years, last quarter number of reportings. And if you see here, the 2021 last quarter, October, November, December of 2021, for the first time, we crossed 20,000 reportings, which is highest and more than any other quarter historically. So this is certainly a very positive observation with respect to data. And this trend also is continuing. We saw very good reporting in January. So looks like for last 1 year, whatever activities are being done at different level, and the noise being created in the system is certainly bringing a lot of energy and awareness within these stakeholders, and things are looking good, but certainly there is a huge scope of improvement from where we stand today. These are the 4 main channels of reporting which the government of India has created. We have an app on Android. We have 1-800 toll-free number. Anybody can pick up the phone and call and put the reporting. And then we take it up and connect with the AMCs in that region from where this call has come, and get the details of the information. We have the ADR reporting forms uploaded on the portal of IPC. This can be downloaded, filled, and again uploaded. And the biggest chunk actually comes from the ADR monitoring centers, which we have created across the country, which I just showed. Currently, this 395 is an old number. We are actually 534 as on date. We have certainly achieved one more recognition, and that is India is 9th largest global contributor to the WHO database when it comes to rank ordering all the members of WHO country who are reporting and contributing to WHO international database. So largest, of course, is United States, and there is a huge gap. But certainly being at number 9 is also not bad. Looking at the state of our social awareness, the education, the systems, all that, being at 9 is not something we should be ashamed of. I'm very proud of this achievement also. But certainly we should try to see how soon we can increase and come at rank order of less than 9, and somewhere between 1 to 9. Certainly in next couple of years, we should be around 5, 6, which I see the trend going on. These are some of the reports which has been generated out of all the data which we collect annually and do the data analysis using a lot of experts, expert committees. Many of the speakers today are part of our expert committees, and I wanted to thank all of them for continued support to PVPI program in meeting our goals and growing year on year. So if you see here till 2020, we had issued 111 drug safety alerts. We had, based on our feedback to CDSCO, 58 package insert changes were done. We actually had reported 9 signals. This is a big achievement from PVPI program. And as all of you know, signals are all those side effects or adverse events which have not been reported earlier by anybody in the world. Last year, we did 18 drug safety alerts based on the data collected. 1 package insert was recommended to be changed, and 3 signals were also detected last year. This is as late as I think 10 days ago or a week ago. This is a press release from CDSCO. There were 11 more advisories sent to state FDAs for the side effects which are coming out of, or adverse events coming out of drugs like ranitidine, IV, ceftriaxone, azithromycin, cloxacillin, itraconazole, ibuprofen, amoxicillin clavulanate, Cipro, dipeptide, DPP4s like sitagliptin, vildagliptin, and saxagliptin, doxycycline, tinidazole. So all these 11 reports also came out of the data analysis which PVPI had done with the help of all the experts, and recommended some changes in the system through CDSCO. So this is certainly an achievement. With all the positive developments, certainly there are some concern area where we need to look seriously, and would like to have support from all the attendees in this program, the speakers, the delegates, the organizers. Help PVPI as you have been doing in the past in addressing the challenges and concerns which we currently have. And the biggest one is the underreporting. If you do a very rough calculation, in a country of 1.3 billion people, we are getting 100,000, around less than 100,000 adverse events. And at any given point of time, not less than 25% people would be taking one or the other medicine. So just think of a situation where more than 30 crores of people are eating one or the other medicine, and adverse events are part and parcel of this whole healthcare system. No molecule, if I can say, is free from side effects. That's part of the property of the molecule or the drug per se. Somewhere it is more, somewhere it is less. But out of 30 crores of people eating one or the other medicine, getting less than 100,000 adverse event reportings is certainly a concern, and certainly an area which needs attention of people like us attending this program today. So I actually have myself, since I joined this organization a year ago, I have traveled extensively and interacted with a lot of people who are working in this value chain. And the kind of feedback which I get is sometimes surprising, sometimes looks funny also, and sometimes looks to be a big area of concern. Sometimes people or practitioners say that, why should I do reporting? I'm not paid for it, which is certainly an area of a big concern, that we also have some moral duty as a practitioner, as a prescriber, or as a doctor. And when we say that we are not paid for reporting, that is certainly a concern, because adverse event reporting is part of your job, being a doctor and doing the treatment of patients. If side effects are there, the reporting this is actually a moral responsibility and also an obligation on you to bring this out. And if you see, a lot of people had a negative connotation on the adverse event reporting. They think that if we report, it is a hit on their either personal brand or maybe the hospital brand, because patients and their family members will start thinking that this particular doctor, whenever they do treatment, it causes side effects, or this particular hospital, when you go there, you see a lot of side effects. But we have to understand two very important things here, that this is not because of doctor or the hospital. It's a property of the drug molecule per se. And if we do not report these adverse events, we are actually going against the system in terms of not giving an opportunity of correction to the system. And how does this correction happen? It can happen in two ways, at least two ways. One is, wherever there is an adverse event, and reporting of adverse events make the system aware that yes, this kind of side effects are coming. And regulators can do a proper intervention at the proper time. They can either change the label. In the worst case, the drug may be withdrawn for improvement. And on the innovator or manufacturer side, they may get the opportunity to do the improvement, do the tweaking required, maybe at the molecule level or maybe at the formulation level. But certainly, unless until they are told that these are the adverse events coming from the field, they would not know that what improvement has to happen. So once we report these adverse events, we are giving an opportunity to the players in the field to do the intervention for correcting the adverse events, or maybe making the patients aware. Think of a situation where if there is a particular drug, say XYZ, and it has a serious side effects of orthostatic hypotension, for example, just an example. And if the patient knows that this is kind of side effects which will come, or adverse event which will come, and if he or she is briefed at the time of prescription, the person would be ready for those kind of side effects and can take care when that particular drug is being taken. As against, if they are not briefed and they are not told about this kind of side effect, that may come as a surprise to the patient. They may fall, they may get hurt, and a lot of subsequent chain reaction can come out of that. And the whole family can get disturbed if they are not told and something comes as a surprise. So by reporting adverse events, we actually help the patients also. And at the end, you help yourself also as doctor when you actually brief the patient about the possible adverse events, and the patient is ready for those kind of events. So some of them also say that I don't have time to report because they have different priorities. But we also have to make them understand that reporting adverse events is also a part of their responsibility. There is nothing like prioritization. That's part of the job. You cannot prioritize that I will not report adverse events, but I will keep on writing prescriptions. Good or bad, currently this whole pharmacovigilance program is a voluntary program in this country. But government can one day, if things do not improve, if the culture does not change, government may also start thinking in terms of making it compulsory, mandatory for practitioners and hospitals to do reportings. But that, I don't think that is the best and most optimal situation we would like to be. So till it is voluntary, it is our duty and our responsibility, and stakeholders like all of you present today have a big role to play. A lot of people say, I don't know how and where to report. I was surprised a couple of weeks back that a message came from ministry that NMC doesn't know that there is a pharmacovigilance program of India. I felt like laughing that how can it be possible that NMC people do not know that there is a pharmacovigilance program of India. But anyway, that was a feedback which came. People think that because of the word vigilance in that pharmacovigilance, people also sometimes feel that there may be medico-legal issue when they start reporting adverse events. They have to understand that it is not their fault. It is because of the drug per se. There is no medico-legal liability because of adverse events. So I'm telling you all this because you are the people who are in a position to influence this whole culture. And we need a cultural shift in this country if we have to make this pharmacovigilance program successful. The owner of this program of pharmacovigilance in this country, we have put a target of 100,000 reportings, minimum 100,000 reportings in 2022. And I'm sure the way things are going, this is not a difficult target, but this is certainly a big milestone for the program, which we are running and trying to take it to the next level. So at the end, I would just like to say that changing the mindset of the stakeholder of PVPI, be it doctor, be it patient, be it their family members, be it pharmaceutical manufacturers, regulators, or pharmacy shop owners, and many more of that kind, nurses, pharmacists, who have a stake in this whole value chain. We have to change the mindset of everybody, and they have to understand the importance of pharmacovigilance. And a very good thing which has happened, which I now hear, that pharmacovigilance is part of the course curriculum at the undergraduate level, in certainly medical education. And also I hear that in pharma education also they have put pharmacovigilance as a paper at the bachelor's level, and certainly at the master's level. So all of us have a role to play in this whole game. And nobody has a smaller contribution. Everybody has equal opportunity and equal responsibility to contribute and change this whole system. Government, as I showed, government has created, now I can say, a huge network in this country for collecting the data. A lot more will be done, can be done, and it is being done. But whatever is happening and whatever growth we will bring in the infrastructure and networking of PVPI program, whatever today we have is also one of the bigger ones in the world. 534 centers, no other country probably has. One of the bigger PV data collection networks in the world. So all this infrastructure has been created. But ultimately, unless until stakeholders like us start either changing our mindset or helping others change their mindset about pharmacovigilance, towards pharmacovigilance, this success of reaching to the next level, taking this program to the next level, and ultimate benefit to the patients will not come. So with this, I would end today, this morning, and I'm sure this wonderful program and wonderful speakers, everybody would have a lot of insights at the end to take away from this particular program. All the best to organizers, and thank you again for giving me this opportunity to speak to all of you this morning. Thanks, and over to you, Dr. Nithya.
Thank you, Dr. Ravenchi, for a very, very insightful talk. And you comprehensively covered not just the strengths of what we have in our system today, but also the challenges that we need to overcome. I see a lot of senior faculty teachers, Dr. Tate, Dr. Shirsagar, Dr. Usha. Would you have anything to add, any comments? Dr. Rajiv may not be able to stay for the entire program. So if there is anything that you would like to ask of him, any thoughts?
Yeah, I was wondering,
Yes,
I had a couple of thoughts, couple of questions actually. Is there something that we can learn from the American experience? Why is it that they are reporting so much? Is it mandatory over there? I don't know if it's mandatory. It's not mandatory. How is it? Is it? I mean, what
so, ma'am, it is actually the socio-economic situation. I mean, when you compare between us and US, it's completely different state of socio-economic condition. The education, the awareness, the participation by the normal public. As a society, we are different, and maybe we are two extremes. So we just have to see how do we shift towards US. It may take a lot more time for us, but certainly the path we have to move. And this is not simply, as I said, it is not government alone cannot do it. You also alone cannot do all this. This need to be a mass movement in this country. A lot of basically awareness programs will have to be run at the government end also, the way we do for a lot of national health programs. So something of that sort will have to be brought in from multiple angles, and then we will see something which currently US is showing. So at this stage, it is not possible to, this is not apple-to-apple comparison. We are completely different country and a completely different society.
Thank you, sir.
If I may just ask, sorry, can I?
Yes, please.
Yes, madam, please go ahead.
Please, madam.
Thank you. Dr. Raju, I just was wondering that one of the important things that should more proactively happen is to give feedback to those who report ADRs, and especially in terms of the benefit that the pharmacovigilance program and the reporting is using. The difficulty with the ADR monitoring is that it is very difficult, actually, very difficult to show in real terms benefit,
right?
Because you can show it as in terms of output, like signal and the other various parameters that you mentioned, but that being translated into real term benefit is something more difficult. So maybe a little bit of work there would be helpful. As you know, WHO has this medication safety as one of their very important flagship programs. And they were once asked in a recent seminar as to how do we measure that. And they've really more or less left to each country that whichever way you measure the benefit of the program. So I think that's one area where it may be useful for the program to consider. I don't know if you have any thoughts on this.
Yeah, ma'am. So that's a very good point. And we know, at least from my background of corporate pharma of last 25 years, we know that unless until anything is measurable, it is difficult to show the growth and bring the growth in that, because you have to have something to measure. Unfortunately, currently, when we see the quantitative aspect in the pharmacovigilance program is only in the number of reportings, number of centers, number of signals, but what happens after that is not being quantitated currently. And also, I know that in this world of technology, the way it has advanced, anything and everything can be modeled. I don't believe that it cannot be done. And actually, I have seen a couple of papers where people have tried to model this impact of pharmacovigilance in quantitative terms also. They actually go to the extent of how much safety would have been come because of this kind of reporting and if this was not there, what would come as the adverse event and what is the impact of that on the national health program or maybe at the personal level also. So that's possible. We have started thinking on those lines. Nothing substantial has still been done, but certainly at PVPI Center, we are thinking that we have to start moving in that direction and bring more quantitative at the patient end.
I may give some suggestion here. As you may be knowing that through ICMR, we have these prescription research, which is an activity. And there at present, there are about 20 centers. I plan to expand those. And there the prescriptions are evaluated, assessed to see whether as per treatment guidelines and what could be a potential impact if they are not as per treatment guidelines. And we do look at there if there could be potentially any ADR occurring. So that's, one of a kind of a reasonable actual impact related thing that could be considered. So maybe we could have at some day suitable to the PVPI group, have a presentation from this ICMR activity, and maybe they can, together see how we can work on that.
That's a good suggestion, ma'am. I'll certainly look into it.
One last question in the interest of time. Yeah. Usha, you wanted to say something?
Yeah, it's just that, most of the clinicians, they feel, especially with, oncology drugs and those sort of things, when you have thrombocytopenia and those sort of adverse events. So they feel, it's a routine thing which occurs. So why report something which is routine? They think PVPI and the ADR monitoring and reporting is something like a SUSAR or something which is unexpected or, something like that. So all the common things, they feel it is doesn't require reporting because everyone knows about it. So your thoughts on that?
So, that's certainly a problem. And I always say that adverse event is adverse event. Common, big, small, uncommon. Those categorizations should actually, the communication to them should be that they should not look into the commonality or uncommonality or small or big. They should simply report. Because what happens that when the data comes into the and becomes part of the database, we might get a different kind of observation when we look at a larger picture. So that is why from the AMCs and the people who are practicing pharmacovigilance and are part of this whole value chain, whenever there is an opportunity, this should be actually communicated to the practitioners that they should simply report. Take only a couple of minutes, not more than that. And then rest the analysis and the use of that data should be left to the people who are doing that, because this is a specialized field.
Yes.
That's what we think and we should communicate.
So just one question, last question, Nithya. What is the number of reports that you get directly either from the patient or from the doctor physician?
So that's very, I feel a little shy reporting that, but certainly that's not even 1%. So I'll give you a number of reports coming through our 1-800. So out of 70,000 reports which we collected last year, the report which came through 1-800, which is actually all non-practitioners. They could be family members of the patient or patient themselves. It was some 346 something through 1-800 number. If I include all the channels and see the reporting by non-practitioners or non-professionals, it is less than 1%. Not even, we don't even cross 1,000. So that's an area which is a huge concern. But as I said, when somebody asked the difference between US and us, it's all the socioeconomic situation. Still, the doctors are drivers of this whole program. And then they become decision maker. What to report, what not to report, why should I report, I don't have time, and all these factors come into play. And unless until we shift this whole dynamics towards the patient direct reporting, this is difficult to handle. So if we have to become a country like US in pharmacovigilance reporting, this whole dynamics has to shift from the practitioners to the patient. A simple theory of consumerism, actually, if I can extend it to that. So unless until consumers start reporting about problem, it is very difficult. It is a analogy between manufacturer of potato chips and consumer of potato chips, and doctor and patient. The patient is consumer, and doctor is actually the prescription, you can say, is the production, the material which is to be consumed. So unless until the consumer becomes aware about the problem in potato chips, the manufacturer will never report the problem, because they think that it is their problem. It is their brand will get hit, and that's exactly how is happening in our PV program also.
In the US and in India, are the pharma industry mandated to report to the Uppsala Center or a government?
They are also not mandatory.
Not even the pharma industry.
So pharma industry have the way we have done. Till it is a new drug, they have to report. I'm not very sure what is the time period and all, and whole
4 year, I think.
logistics.
4 year they put that.
But only a part of it is mandatory, not the whole program. Whole data is not collected through.
And even USA it is not mandatory for the pharma to report.
No.
Okay. With that, I would like to thank Dr. Raju. Thank you so much for coming and talking to all of us. We have a large audience on YouTube listening in, sir, and we will send you the report of the attendance at the end of this. Thank you for your insightful presentation, the comments, and I hope under your leadership and with all that you've said, as a country, we will be able to achieve this target of 100,000 ISCRs, as is your vision. I would also like to thank Dr. Thatte, Dr. Shirsagar, Dr. Usha for their comments. Yeah, thank you very much. And I would request Dr. Mahesh to take over and introduce the next speaker and go ahead with the session. Thank you so once again.
Thank you. It was a pleasure.
Okay, ma'am. Thank you, sir. Thank you, my senior teachers, for the discussion. Our next speaker is Dr. Nilma Shirsagar. Madam, I welcome Madam for today's program. Madam is presently Chairperson Core Training Panel, Indian Pharmacopoeia Commission. Previously, Madam was Professor and Head of Department, Clinical Pharmacology at Seth GS Medical College, and Dean and Director of Medical Education and Research, Seth GS Medical College and TN Medical College, Mumbai. Madam is a former National Chair, Clinical Pharmacology, ICMR, and Emeritus Scientist, Basic Medical Sciences, ICMR. I request Madam to proceed with her talk on Introduction to Core Training Panel. Madam, please.
Thank you very much, Mahesh, and thank you, Nithya, for organizing this very well thought about program. Since we are running a bit behind schedule, I won't take much of the time. I don't have any PowerPoint presentation. And I would just like to update a bit onto the core training panel, what work is being done through that. As we just heard from Raju, the entire scope of the pharmacovigilance program is what we have heard from him. And as he said that there are various groups of or various sources from which we get the adverse drug reaction reporting. So when it comes to the training panel, as you can well imagine, that in order to encourage these various so-called stakeholders to report, it's very important that a good communication is given to them, both for reporting as well as giving them a feedback about what their reporting means in an overall context. So that's the job of the core training panel. And there are various training programs which are organized, partly these are organized by the center itself, where they provide training to new entrants and further updates and further advanced level training. The ADR monitoring centers themselves are expected to conduct training programs. This one is one of the very advanced level training program, as we can see from its content. One of the important things which we have always been discussing, and where we find that it is not being implemented across the various 500 odd ADR monitoring centers, is being able to go out of their own institutions where the ADR monitoring center is located. And several suggestions have been given, such as being able to hold these training programs along with the local IMA branch or any other clinical discipline branch, so that we can expect from private practitioners and practitioners across their own area of jurisdiction of activity. So that is something which is important. The other important thing is to be able to include lay people or patient groups. And more recently, through this training panel, there have been suggestions, and these are being worked out. As you know that we now have Bijan Mishra, who is heads the consumer activity, and also we have Vivek Ahuja from a private sector. And so in terms of the content and also in terms of the delivery, so that the target audience, accordingly the content and the delivery has to be modified so that we get the desired effect. So that's a sort of activity which the co-training panel is at present into. And all of you have contributed already quite a lot on this kind of PowerPoint presentation, sets of them for general training and the content of it. We need to move on from there and see how these can be expanded. One could I would just like to mention here African proverb, which says that in order to bring up a child, it requires the whole village to contribute to it. And so rightly said. So if we want to have safe use of medicine, we need contribution from all the stakeholders, from the practitioners to the patient to the pharma industry. So the part of this training panel is therefore to see that all of these stakeholders are addressed in the training programs. The program that we have today is very fantastic and fascinating further output that we can expect from the pharmacovigilance program. I mean, I can see excellent speakers and excellent topics, right from risk benefit to public health. It's important to also recognize that all ADR monitoring centers need to be able to connect to the public health programs. Jan last year, when the vaccines were introduced, we had 2-3 training programs, which were with the AEFI, and particularly relating to COVID vaccine. Now, one crucial thing is that this required a very proactive and very sort of a quick response, like an emergency response. So I would say that in order to be able to cope up with such emergency response, because now the demand on pharmacovigilance has taken a completely new shape compared to 2 years back. Pharmacovigilance program used to be a bit of a rigid program with very well-defined structure and all that. The whole thing has changed, become completely topsy-turvy, and it has put a lot more demand on pharmacovigilance. So the ADR centers and their trainings have to be accordingly, suitably updated and modified. So these are some of the thoughts and activities that I would like to mention about the training panel here. And thank you once again, Nithya and your team, for organizing this excellent program and for inviting all the experts, including Dr. Usharani. And I could see that you have international speakers. Our friend Dr. Robin Perner is there, and also Tanu. And you also have Dr. Arora, who is heading at the moment Government of India's vaccine-related decisions. So thank you, Nithya, and I hand over back to Mahesh. Thank you very much.
Thank you, madam. Thank you for your talk on co-training panel. Now we all became more aware of the various activities of the co-training panel. Now I request Dr. Nilima Shirsagar madam and Dr. Raju Singh sir to proceed with the next scientific session as chairpersons and introduce the speakers and begin the proceedings of this scientific session. Thank you.
Madam and sir, just one thing. Dr. Tanu will speak first. She has an OPD at 11 o'clock. So she will start and then I will be the second speaker for the session.
Okay. I have to a bit apologize because now it has got a bit complicated and I have to be at least present for a while into another meeting. So I would just request that the next session, if I'm not there, for Nithya to kindly, also monitor, or Mahesh to monitor the discussion that follows after the talks. So great privilege and pleasure to introduce Tanu, who is a consultant pediatrician at the Kokilaben Hospital. And she is one of the well-known speakers and she is a very well-recognized expert. And it is wonderful to have a clinician pediatrician into this program. And that sort of should be able to bust some of the myths that, or actual some facts that Raju mentioned. And she's going to speak on benefit risk analysis for repurposed drugs, such an important area, and as a clinician's perspective. So I hand over to Tanu then. Tanu.
Thank you.
And pleasure seeing you.
Thank you, ma'am. And I would thank Nithya also for inviting me to speak this time again. I did it last year. And with that, I'm just trying to share my screen. So my screen is visible?
Yeah, Tanu, put it in full screen mode and you're good to go. Yeah.
Thank you. So basically, we will be, to start the topic, when the COVID-19 pandemic hit us in beginning of 2020, 2 years ago, there were no treatment options, and understandably so, because this was a new virus. Therefore, because it was some treatment was needed because patients were falling sick, repurposed drugs were used. And this use of repurposed drugs was on the basis of various principles. One was on the basis of experience with the previous SARS and MERS virus, and also with other RNA viruses. Then on the basis of understanding of the virus life cycle, or the pathogenetic pathway, because it was soon recognized that the virus causes a cytokine storm, so anti-inflammatory drugs were used. Drugs like lopinavir, ritonavir, and remdesivir were used because they had been used earlier with Ebola, MERS outbreaks. Also on the basis of some in vitro data where they looked at the effect of the various drugs on the virus, and also on the basis of small observational studies which came within a couple of months of the pandemic starting. So treatment was initiated, and at the same time, randomized trials were also started to evaluate the effect of some of these drugs. But then, as the results of the randomized control trials came in, several drugs were actually abandoned. And but still, most of the drugs that are used against COVID-19 are still repurposed actually, and we'll discuss them. And new drugs against COVID have been few, and we can count them on fingers, and I'll come to them later. And in fact, there is a lot of work being done in identification of new targets and new drugs by artificial intelligence and machine learning. So let us just finish off these drugs now, because these were repurposed drugs which really shot to prominence 2 years ago, but now we know that they are no longer efficacious. In fact, hydroxychloroquine is the first on the list, And I remember using it in patients in the first 2 or 3 months, and all of us as healthcare workers in Kokila also took weekly hydroxychloroquine based on some reports. But then soon it went out of fashion. Then came azithromycin, or simultaneously with hydroxychloroquine, because there were a couple of friend studies which showed that azithromycin brought about reduction in the viral load. But then it was also given up because later trials didn't show benefit. But unfortunately, azithromycin is still used a lot in patients with COVID-19, despite there being no evidence of benefit. And that has actually compromised this drug, which is so important for management of typhoid fever, lower respiratory tract infections, and bacillary dysentery. Lopinavir ritonavir was used on the basis of the use in SARS and MERS epidemics, but soon there were a couple of good randomized control trials, and also the Solidarity trial, which didn't show any benefit, and it was given up fairly quickly. Ivermectin and doxycycline are drugs which actually were not commonly used in the first wave, but were widely used in the second wave because there were conflicting results about the efficacy of ivermectin. And in fact, but then now more and more evidence is accumulating that ivermectin is not useful. Interferons were also evaluated, fairly expensive drugs, but later on given up. And fortunately, these were not used a lot in India. Convalescent plasma was another drug which was used in the beginning, and then there was a kind of run for convalescent plasma. People were running from pillar to post, but then the ICMR trial and many other RCTs came in which showed that convalescent plasma is not useful. So also methylene blue and vitamin C and zinc are used a lot in COVID-19, but we know that actually they have no benefit. Then we come to repurposed drugs of uncertain efficacy, where actually the final word has not been said yet, but more or less we know that they don't do great, and that includes favipiravir, which was used a lot after the first wave and in the second wave. Colchicine, for which there is conflicting data, RCT showing benefit, another one showing none. Fluvoxamine, which is an SSRI, again of uncertain efficacy. Vitamin D data, both for and against, used widely still, sometimes leading to toxicity. And then itolizumab, which was our own indigenous anti-CD6 monoclonal antibody, which was used when tocilizumab was not available. We have one trial showing some benefit, but a lot of side effects. And then we have those drugs with a favorable efficacy profile. And these includes both repurposed drugs. And in this list, I put anticoagulants, remdesivir, steroids, IL-6 inhibitors, and JAK2 inhibitors. And among the new drugs is molnupiravir. Actually, molnupiravir was developed for influenza, but it was not really clinically evaluated for that. So I put it in the new drug group. Nirmatrelvir or Paxlovid, and monoclonal antibody cocktails. And those which are marked with an asterisk are the ones which have been approved by the World Health Organization. So in this list, I'm going to discuss these 5 drugs because these are the ones which are most commonly used in the management of COVID-19. So to start with remdesivir and its timeline of use. So it is a repurposed drug originally developed for Ebola virus disease. And the drug, when it was used, it's contraindicated in patients who have liver dysfunction or low creatinine clearance. And the use of remdesivir actually started on a compassionate basis in Feb 2020 in USA. Then the company embarked on the Act 1 trial, and this was a randomized controlled, placebo-controlled, double-blinded trial, which showed that 10 days of remdesivir brings about faster recovery in patients with COVID-19 as compared to placebo, but there was no significant impact on mortality. And in India, we started using it in July 2020. Sorry, that should be July 2020 when it was made available. And then came this WHO Solidarity trial, which showed no benefit with remdesivir. The ICMR also recommended against unrestricted use, but it was used widely through July 2020 onwards into the second wave. And there was a serious shortage seen during the second wave, and also spurious drugs. So because this is a pharmacovigilance meeting, in April last year, there were a lot of these newspaper articles which were showing indiscriminate use, hoarding, and black marketing. In fact, a single vial which cost 5,000 was selling at 50,000. And also there was, spurious drug being sold in, in fact, glucose salt solution being marketed as remdesivir, which is really unfortunate. And also another thing which we forget was that, in this hurry to manufacture the drug to meet the requirements, sometimes there were shortcuts, used. And because of that, many patients who got remdesivir got shivers, fever, rigors because of endotoxin in the drugs. And this was by many companies. So that was also a problem, which was not inherently due to the drug, but because how it was manufactured. So assuming that the drug is proper, then what is the evidence about the efficacy of remdesivir? So I'm not going into individual trials because there are so many. So I've picked up this meta-analysis, which was actually done by AIIMS New Delhi, in which they looked at the 4 major randomized control trials and the effect of remdesivir on mortality. And because the largest numbers were from the WHO solidarity trial, which didn't show any benefit, actually this there was no mortality benefit seen with remdesivir in this meta-analysis. However, when you look at clinical improvement or time to improvement, you find that there was benefit which was seen. Because here, actually, if you see, the WHO solidarity trial was not included. And we are more interested in the serious adverse events. So they actually see, now one more thing, that most of these trials, when they define a serious adverse event, it can be disease related or it can be treatment related. So if the person progresses, the respiratory failure progresses, or the person ends on a ventilator, it is also counted as a serious adverse event. So, also you find that actually it is in favor of remdesivir, and that is partly because of the prevention of progression of disease due to remdesivir, and it also tells us that drug-related adverse events were uncommon. Then this is a relatively recently published trial which was done way back last year, but here they use remdesivir early in the disease. Because it's an antiviral drug and it should be given early to produce good outcomes, unlike the previous trials which used remdesivir when the person had already become hypoxic, where the role of antiviral drugs is less and the role of anti-inflammatory drugs is more. So in this Pine Tree trial, where they gave remdesivir to unvaccinated adults and it was given for 3 days in non-severe COVID-19, they were able to show a reduction in hospitalization and death by 87%. Also, this trial gave more information about the adverse effects of remdesivir, and where they found that though there were slightly more adverse events in the treatment group as compared to the placebo group, the effects were usually mild. And also the biggest concern about use of remdesivir was its effect on hepatic and renal function. And in this trial, actually, they didn't see any significant renal or hepatic dysfunction due to remdesivir. We also have this study from CIPLA, where they reported the safety and clinical outcomes of about more than 2,000 patients who received remdesivir in 2020. And they reported about 119 adverse events in this cohort of about 2,800 people. And the most common adverse events were nausea, vomiting, increase in liver enzymes, bradycardia, nephrotoxicity, etc. But because there was no control group, we cannot say that how many were causally associated with remdesivir. And this finding of sinus bradycardia was actually also reported as a safety alert by the Indian Pharmacopoeia Commission. And that is also one of the reasons why it is recommended that remdesivir should always be given in hospital under supervision and not at home. What about our experience? We have used remdesivir extensively in more than 2,000 patients, even in preterm neonates, and we've really not noted any significant adverse events. I mean, we had seizure in one patient where we had to stop it. We've had severe nausea and vomiting in a few patients where we had to stop using remdesivir. And whenever we found that the liver enzymes rose, we gave a day's break. And after they came down, we restarted remdesivir and we were able to finish the course. And because we saw good benefit with remdesivir, especially when we used it early, and this was later confirmed by the Pine Tree trial, we used it even in patients with renal failure, on dialysis, and with hepatic dysfunction. And gradually it is being recognized that if it is really indicated, it can be given in patients with renal failure, because remdesivir as a drug has low affinity for the mammalian RNA polymerases in the kidney tubules, and also the cyclodextrin carrier, which accumulates in the body if there is renal dysfunction, at the dose in which it is used in clinical practice is not a problem. So also we have publications talking about the safe use of remdesivir in patients with renal dysfunction. Moving from remdesivir to corticosteroids. Now we started using corticosteroids at our center 3 days after our first patient came in. That means way back in May 2020. And this was based on experience at the Henry Ford Hospital, USA, who were already well in the first wave, and they were using corticosteroids. And on an informal conversation, they said that it was helping their patients. And also there were some reports from China showing benefit. Though we were using it off-label, the recovery trial, which published its results in June 2020, showed mortality benefit with corticosteroids, and therefore other ongoing trials with steroids were prematurely terminated. And then corticosteroids became the standard of care for hypoxic COVID-19. And the dose which was recommended was 6 milligram dexamethasone or equivalent for 5 to 10 days. And there is a recent trial which has compared 12 and 6 milligram, and it reported better outcome with 12 milligram in patients who are critically sick. So again, I chose the World Health Organization meta-analysis on use of corticosteroids in COVID-19. And again, as you can see from this forest plot, that there is benefit with corticosteroids and 28-day all-cause mortality with approximately 30% reduction in mortality, and the upper bounds of the confidence interval is just exceeding 1. Similarly, the trials also reported serious adverse events, except for the recovery trial, which didn't report on adverse events. The other trials reported on adverse events, and here also you can see that there was it favored the steroids, and this is possibly because of the fact that many of these adverse events are due to disease progression. So therefore, if you prevent disease progression, you reduce the serious adverse events. And in fact, you even reduce infections because your patients are in the ICU and on ventilator for less duration of time. Now, the other important thing which I would like to highlight is that what about use of steroids in non-oxygen requiring COVID-19? And this is again a meta-analysis published by AIIMS New Delhi. And what they found was that if you use steroids in patients who were not hypoxemic, there was no benefit, in fact, in progression to severe disease. In fact, the steroid arm showed greater progression, and in fact, the steroid arm also showed greater mortality. And also the corticosteroids did not significantly impact fever duration, viral clearance, or hospital stay. And that is why it is so important to not use corticosteroids in patients who are not needing oxygen. But steroids were used rampantly in India, and more so in the second wave when hospital beds were scarce. They were used in non-hypoxemic patients, and when indicated, they were used in very high doses for prolonged periods with inattention to sugar control. And many people who got steroids didn't know that they were diabetics. And the most serious outcome of this rampant use of steroids in the second wave was the epidemic of mucormycosis, which affected more than 40,000 patients and caused more than 10,000 deaths. In fact, in a study by Patel et al., which was published in Emerging Infectious Diseases, they found that in the patients with COVID-associated mucormycosis, in 33% steroids were given when not indicated, and in 64% of the patients, high or prolonged doses were used. The other adverse effects associated with steroids, which we have seen, are high blood sugars, which make it very difficult to control, GI bleeding, and also a predisposition to other nosocomial infections. And While the trials with steroids did not see an increased infection rate, it may be because they used steroids at a proper dose for a proper duration in a setting where there was very good infection control. And this is just to tell you about what happened to our antimicrobial use when the COVID pandemic stuck. So this was, this is a percentage of patients who are prescribed restricted antimicrobials, which includes colistin, tigecycline, echinocandins, etc. And you can see that this is data for Jan, Feb, March before COVID. And when the first wave came, you see a sudden rise in the prescription of antimicrobials and percentage of patients who are receiving antimicrobials. And then that curve tended to come down as the number of cases decreased. And again, with the second wave, we saw such a huge peak. And the use of restricted antimicrobials was 3 times the previous time. And this is because of the fact that a lot of patients who had been staying in hospital for a long time ended up with secondary infections, which necessitated use of antimicrobials. And some of these infections may have been contributed by steroid use. Coming to the IL-6 inhibitors, which includes tocilizumab and sarilumab. Sarilumab is not available in India, and therefore I'm not discussing it. It pertains to tocilizumab. And it was introduced fairly early in the pandemic when they recognized that there was a cytokine storm with high IL-6 levels. And we used it in March 2020. After that, there were several observational studies which supported the use of tocilizumab, and its use picked up in the first wave. It led to intense shortage of the drug, even drove use of the subcutaneous formulation. But at that time, there was no clarity on the use of tocilizumab. So it was being used in high doses, 8 mg per kg, 2 to 3 doses, and even being used late. But then we had the investigator-sponsored trials which showed no benefit and the use declined. But then came the recovery and the remap-cap trials which showed benefit with tocilizumab. And WHO approved the use of tocilizumab in July 2021. And the use of these IL-6 inhibitors increased towards the end of the second wave in India, where people were more rational with use of tocilizumab. It was used early and in truncated doses. But at the same time, because it was needed and it was not available, there was spurious products in the market. And in fact, this was just a recent notification saying that, on the complaint of the company, tocilizumab vials were analyzed and they were spurious. So imagine, a cost of a vial, which is about 43,000, and giving a spurious drug. So again, I've chosen the WHO meta-analysis for tocilizumab, and it clearly shows that based on so many trials, of which one, of course, was conducted in India, that there is a mortality benefit with use of tocilizumab if it is used early and it is used in patients with high inflammatory markers and it is used alongside steroids. And one great concern with use of tocilizumab was the risk of secondary infections. And again, when these trials looked at the risk of secondary infections, they did not find an enhanced risk, at least in the trials. Now, what about India? So there is concern about secondary infections that may not have been captured in the trials. And you have one trial which was set in India, which also showed no difference in infection rate between tocilizumab and placebo. But we must remember factors which are operating in the Indian setting, which may not be operating in other countries. One is suboptimal infection control practices, high background resistance rates in gram-negative pathogens, and suboptimal timing depending on availability, because you actually need to give the drug within the first 24 to 48 hours. And if it is not available, if it's available after 3 to 4 days, it's given at that time, where actually it may not benefit the patient and it may actually cause more harm by increasing the risk of secondary infections. And we are still awaiting observational data from India regarding the adverse effects of tocilizumab. But in the KEMH study, they found that only 2.7% of the COVID-associated mucormycosis patients had received tocilizumab. So possibly it didn't really contribute to that mucormycosis outbreak which happened. Last is the JAK2 inhibitors, which includes baricitinib, which I'll be discussing further. And again, you have a recent systematic review which has analyzed 3 major trials where baricitinib was used. And as you can see that baricitinib gives mortality benefit over steroids, and therefore the WHO has also recommended addition of baricitinib to the background regime of steroids with or without remdesivir in patients who are hypoxic. And again, there was a concern about adverse events with baricitinib because, and you can see from this that actually they have not, there is no significant difference in the adverse events, serious adverse events or infection rates with baricitinib as compared to placebo. So because baricitinib has been used in rheumatoid arthritis, we have that data also available about its safety. And there they have said that there is a little higher risk of infection in those treated with baricitinib as compared to placebo. And the risk of severe adverse events was higher with the 4 milligram as compared to 2 milligram dose. And there have been concerns about venous thromboembolism and herpes zoster with baricitinib. But at least in the COVID trials, you have, when you go through the studies, they've described the adverse events in detail with adjudication about whether they were due to disease progression or drug related. And they have found that even treatment related adverse events were lower in the drug group. And the incidence of infection was lower with baricitinib in the first trial, which could be due to faster recovery and less nosocomial infections. And therefore, the difference which you see between rheumatoid arthritis and COVID could be due to the duration of therapy, because in COVID, we give treatment which is for 14 days or less. And we are still awaiting post-marketing surveillance data, but we've used it since July 2021 and have not noted any serious safety signals. And finally, to talk about molnupiravir, which is actually a prodrug of NHC, and its mechanism of action is that it causes mutagenesis in the virus, leading to replication errors. And animal studies with molnupiravir have shown that it is beneficial, but it has also shown cartilage and bone toxicity if given for prolonged periods in high doses, and also embryo toxicity when given to pregnant rats. And therefore, the drug is not approved for pregnant women or children. There are also some concerns about mutagenicity because if it can cause mutagenic effects in the viral RNA, it can also cause in human DNA. But you have these two papers doing the rounds, one which says that it is mutagenic, and then there's a rebuttal from Merck which say that they have done a lot of experiments in animals and other cell cultures, and they have not seen any evidence of mutagenicity. So this trial, which looked at non-severe COVID-19 in unvaccinated adults at the time of the Delta wave, it showed that if molnupiravir is given within 5 days at a dose of 800 milligram twice daily, you have a 30% risk in hospitalization and death. And actually, it's significant to see that there were 9 deaths in the placebo group as against 1 death in the treatment arm. And again, there was no significant difference between molnupiravir and placebo as far as the adverse events are concerned. So the drug appeared promising, but remember, it was in the Delta wave in unvaccinated adults. And also, they didn't see the benefit across all subgroups. So, and the number needed to treat, which I think Nithya is going to talk later, to prevent hospitalization was 33 and to prevent death was 83. So relatively high NNTs. So molnupiravir protected against death and therefore and hospitalization. So it was approved by US FDA, UK and Indian DCGI with these caveats. But at the same time, we have to remember that there is a cost and pill burden, and there is also that potential risk of mutagenicity. Also, in the current scenario where you have a less virulent virus and you have high population immunity, the NNT will rise even further, and therefore the benefit of the drug is going to decline. Therefore, if we are using molnupiravir in the current setting, we have to select our subjects carefully, chiefly either those who are unvaccinated or who are immunocompromised, who have risk factors for disease progression, and try and avoid it in people of the reproductive health group, because in those anyway, the risk of disease progression is lower, and the if there is a mutagenic effect, they have a long life ahead of them, and they may show up effects later. We have used it in very few patients, but have had some defaults because of the pill burden. And also in the Omicron wave, people got better on the second day. So then they didn't feel good enough about taking the drug for 5 days. And many of them stopped treatment after a couple of days. So this is a drug which is of limited use at this time, but maybe of benefit if there is a more virulent variant which comes around the corner. So finally, to end, I would say that for me as a clinician, COVID-19 has been a huge learning experience. It's been like a once in a lifetime experience, and I've seen so many therapies which have come and gone, and a few have stood the test of time. And overall, I would say that the therapies have been safe if used correctly. But India has seen tremendous misuse of certain drugs, like HCQ, azithromycin, ivermectin, doxycycline, remdesivir, antibiotics, and steroids. And even now, once you have clear data showing that many of these drugs are not effective, prescription of some of these drugs continues, which is actually a cause of worry. And also the collateral damage because of this, which means increased cost of therapy for the patient, adverse effects, and also increased antimicrobial resistance because you've used so much of azithromycin, doxy, and other antibiotics. But I would like to end at a positive note. I would like to say that the pharma industry of the country has been really up and about. And although there were those instances of drug shortages, but by and large, I think we've fared better than many other countries, which, saw a paucity of drugs as simple as oral steroids. I think with that, I would like to end and hand it over to Dr. Nithya.
Madam is there, Dr. Shirsagar is there, and Madam, I've requested Dr. Thatte to step in as well. Yeah. Madam, you're on mute. Yeah.
Yes, thank you, Tanu. And you have very rightly brought out that it's a huge learning experience, and that for various drugs, they fell out of, by the wayside, so to say, with randomized control trials and the results of that. And the bottom line, as you very correctly mentioned, was that this required, the drug required to be used in the right doses and at the right times period in the COVID overall disease progression. Also, there's something like, observational studies which were also put into. We had done at the ICMR study with the hydroxychloroquine in healthcare workers, but as pre-exposure prophylaxis. The treatment part has shown non-effect, but pre-exposure prophylaxis, lots of studies did show benefit. And but now with vaccine, obviously, it's not that relevant or applicable. But I think it's still important to keep that data with us, maybe required future. But if you look at the benefit-risk benefit, you have very correctly pointed out the risk of inappropriate use of medicines. And great, I will just request Nithya Omila to now see if there are any more questions from the audience.
So there is one question which I can see here, which I think Mahesh has or Sukant has put forward. I mean, the question is from a delegate. Since there were no guidelines for the use of steroids by the government, even though trials were there, we ended up in having too many deaths due to steroids' adverse effects. Your comment on that?
No, I think there were guidelines from the government, because as soon as the recovery trial came out, the ICMR included steroids in its guideline, and it very clearly said that it is not for patients who have no hypoxia, and it has to be used in that particular dose for this much of time. That was set out just on the heels of the recovery trial. So I think those guidelines were there, but unfortunately they were not adhered to by most of the practitioners. So that was the reason. I think unregulated use was the reason for the deaths which could have happened because of steroid adverse effects.
Which leads me to ask you a question, Tanu, is how can we change this behavior of physicians? What can we give them as an incentive to change their behavior to do more rational prescribing? Because I have, in my whole career as a clinical pharmacologist, I have experienced tremendous resistance to listening to anybody other than their own experience, which they think is superior to everything else.
Yes, so that is a problem, and it's not only with steroids, it's with antibiotics as well, all those things. And so I really don't know, because there is nothing like, some punitive behavior associated, or punitive, action, sorry, to this kind of behavior. Because and it was not that people were not being educated. I mean, there was education from the lay media, there were the guidelines which were coming out, guidelines from professional societies, guidelines from COVID task force, webinars which were talking about it, but people were not following them. So I really can't say what can be done. Actually, what really changed the behavior was after the COVID associated mucormycosis. After that hit, the patients themselves became very, very careful. And in fact, though the media called it black fungus, but it really changed the way, people use steroids after that. Because then you could clearly see that, the fear of God was in both the patients and the doctors, and the use of steroids came down. But that was a bit too late, I guess.
Thank you, Dr. Tanu. I think we can, Nithya, should we move on now?
Yes, madam, please.
Thank you, Dr. Tanu. This was a very educative talk. I enjoyed myself thoroughly. I can see Dr. Rakesh Agarwal is also on the program. And as we just talked, I will introduce Suparna, Nithya's talk will come, but it will come a little later because Suparna also needs to leave. So she will be doing the session now. And since we have Dr. Agarwal with us, we should take that session now, which we will do. So Suparna is a professor of pharmacology at the Institute of Postgraduate Medical Education and Research in Kolkata. And she's a very important member of my signal review panel of the Pharmacovigilance program of India. She contributes tremendously to this program. I have to say thank you to you, Suparna, for this. And apart from a degree in pharmacology, she also has a degree in dermatology. So you're here as a clinician as well. So you need to also tell us about why people don't report adverse events. She's a prolific writer with a keen interest in Indian regulatory guidelines and antimicrobial use and resistance. So I will now hand over to Suparna for her talk on principles and methods of vaccine signal detection.
Thank you, madam, for the kind introduction. Thank you, Nithya and Indian Pharmacopoeia Commission for giving me this opportunity to share some of my knowledge in this regard. But I'll be very brief and will lay down the main principles of signal detection with a special focus on vaccine-associated signal detection methods. So I'm going to share my screen. 1 minute, please. Is my entire screen visible? Ma'am, is my screen visible?
Yes, it is visible. Make it into a slideshow.
Okay, ma'am. 1 minute, ma'am. It is already in slideshow.
It is not right now in slideshow.
Now is it in slideshow?
No, no.
It's not. Just a minute, ma'am.
Yeah.
Is it now in, can you see it now, ma'am?
No, it's not in slideshow. I can see your slides, but it's somehow not converted to
So
slideshow.
should we say that her fonts are fairly big and readable, so we stick to this?
Okay. Yes. I think if it's readable, then can I proceed? I think there's some technical.
Yeah, go ahead.
Okay. So in the next 20 minutes, 25 minutes, I will essentially speak on the principles and methods of vaccine safety signal detection. And this is how I have outlined my talk: background, general approaches, some disproportionality analysis measures. Vaccine.
No, we haven't moved to the second slide, Suparna. We can only see the first slide still.
Okay, ma'am. Sir, just a minute, Dr. Agarwal. I think there is some technical glitch. I'll just sort it out 1 minute.
Yeah, or you send it to Nithya and they can project possibly.
Okay. I'll just mail it to her.
Yes, please. I'm online. I'll just share it from my desktop.
Okay. Nithya, shall I send it to your Hotmail or your both?
Yes,
KEM?
my Hotmail and both. Yeah, they are routed into each other. Not a problem.
Yeah. I've sent it, Nithya.
Yeah, just give me a minute.
Then I'll going to stop sharing. No?
No, I'm co-host. I should be able to share. Just give me 1 moment. Suparna, I haven't got it yet. Maybe it's a large file. Yeah, just wait for it to come.
Okay. I'm trying once again.
Yeah, just try once again. It's not yet come. As soon as it comes.
Entire screen, just 1 minute.
Yes, Suparna, I've got it now. Let me just download it. Are we okay now?
Yeah, it's a full screen.
Yes, Suparna, please start. I'll change the slides as in when you tell me. Suparna?
Yeah, can you hear me now? Yes.
Yeah, please go ahead. Yeah, I'll change the slides. Yeah.
Yes. Next slide, please. So this is the outline of my talk. I will be speaking on the background, then on the general approaches, some disproportionality analysis metrics which are used for signal detection, some special issues pertaining to vaccine-related signal detection, a brief couple of new analytical tools which have come up, the caveat and the take-home message. Next slide, please. So in order to set the scene, we all know that immunization is the most cost-effective public health intervention in preventing various communicable diseases. But next slide. But we always know that vaccine safety issues have always made headlines, whether it is the print media or the electronic media. We will see that there is a tsunami of information which is coming up, including in scientific literature regarding vaccine safety issues. And therefore, this reiterates that all stakeholders need to be very transparent and critical in analyzing and interpreting vaccine safety issues. Because you're always, unlike small molecules or the conventional drugs, this is far more under scrutiny. And therefore, we need to define what a signal is. A signal is essentially trying to identify unknown or less commonly known risk with medicine use. Next, please. And it is essentially a reported, how is a signal defined? It is reported information on a possible causal relationship between an adverse event and a drug, this relationship being unknown or incompletely documented previously. So I have underlined certain important, the previous slide. Yeah. So I have underlined certain important aspects of the definition is that this is a possible causal relationship and between an event and a drug. In this case, it may be a vaccine. But this relationship or some aspect of the relationship is unknown or incompletely documented. That means either the nature of the adverse event is unknown, or it may be the frequency at which it was known, now it has changed, or the severity that which was not known, now it has changed. So it is either unknown or it has been incompletely documented. Next slide, please. So the important characteristics of a signal is that we must remember that it is a hypothesis of a risk, and with the medicine, and the latter part of it is extremely important with the data and arguments that support it. So it is not merely trying to do a clinical review or coming up with some statistical metric jargon. It is whatever communication and outcome of your signal detection takes place, it has to be supported with the data and arguments. And very often we find that the evidence that we have at the point when we are doing a decision making may not be conclusive. And therefore, this reiterates that you need to continue to follow up this and to see what trends, because we often find that what may not appear to be a signal at this point of time may become a signal with certain more data coming in or following reanalysis. Similarly, what may appear as a signal today may die down and may not be considered as a statistically or a clinical signal in the long run and on the latter point of time. And it is the main importance of it essentially lies that this is an early indicator of some risk. And it often entails, if you see important communication, say, for example, the European Medicines Agency, the Pharmacovigilance Risk Assessment Committee, they often, while doing signal detection, when they are unable to come at a conclusive decision, they often request for providing to the sponsors or the manufacturers, marketing authorization holders, some additional or new information. And the main ethos of signal detection is accuracy as well as timely detection, because accurate detection of a signal or accuracy is extremely important so that we cut down any spurious associations or false positive association. And this is all the more for vaccines. And timely detection, that is also exceedingly important. If there is too long a gap or a lag between detection of a safety signal, this has a tremendous impact on the healthcare providers and to the consumers or the patients themselves or the volunteers or the healthy population who are essentially taken. And of course, it is also important that this early detection will benefit the marketing authorization holders and of course the regulatory authority and policymakers as well. Next slide, please. Now, with regard to vaccine safety detection, there are some special issues, because vaccines are essentially a large number of vaccines, whether it is pediatric vaccine or adult vaccination or immunization programs. The impact, this is because of public health program, its impact is far more than the small molecules or the other drugs which we use. And therefore, you have to be very critical in evaluating the risk versus benefit, whether it is clinically important, what is its public health impact. So here you have the dual role of assessing the safety impact in terms at an individual level as well as at a community level. And of course, at the background, you always remember that there is a proactive role of media and activist groups, especially with regard to vaccination. So these are some special issues in vaccine safety signal detection. Next slide, please. Therefore, the approaches in signal detection are essentially, basically, these are either qualitative and quantitative approaches. Now, the qualitative approaches which we see is you have a descriptive overview. And in this descriptive overview, you do not look into the maths and science or statistics of it. And this is, it was and it shall continue to remain as the most effective, one of the most effective measures. And it is not going to be surpassed by any statistical tool. So you need a multidisciplinary team who will look in threadbare into the narratives which are present in individual case safety reports or in the medical literature, and they will try to find out whether what is being reported or whether whatever is available in the literature, scientific literature, essentially mandates occurrence of certain unknown or less frequently known documented safety issues. Next point, please. So what do these, in qualitative review, what do we do? Essentially assess the quality of information. And this assessment of quality of information can be done for individual reports, that is in a database of spontaneous reporting system, we often say ICSRs, that is individual case safety reports, or it can be in the literature, it can be simple case reports or case series, or you can do an aggregate analysis of aggregate of reports which are present in a database. And often what we need is that we need to also review additional information when we are doing qualitative reviews. This additional information review essentially entails looking at literature, other regulatory authority mandates and communications, and even asking for more information from the manufacturer or the marketing authorization. But one of the limitations and disadvantages of a qualitative review is that it is essentially time consuming. It is not feasible as an initial screening tool when you are hunting or looking at a big database. And therefore, as a result of which this is often substituted, or I should say, it is the hand-holding of the initial screening tools are essentially now the quantitative methods along with the qualitative ones. So what exactly is the quantitative approach in signal detection? The quantitative approach in signal detection essentially entails that is fishing out or screening of a data from a large database. And these databases can be of several types, and I will talk about it in my next slide. And after you sieve and look at the data, you do a statistical analysis. And therefore, these quantitative measures are essentially useful when you're looking at a large database, or when you're looking at, trying to look at important health, electronic health records. So where you have a lot of data and therefore you need automated tools to try to find out as to what is being reported and what you expect to occur. And one of the important outcomes or the metrics of any quantitative approach analysis and signal detection is known as disproportionality analysis. And I will talk in details about it in the next few slides. But the basic ethos of a disproportionality analysis is to see whether the observed reporting counts are more than the expected counts. So that is the basic principle of detecting a signal using the disproportionality metrics. Next point, please. And of course, this has become very attractive because these are automated devices and you have very quick deliverables. So you have softwares in place, and say, for example, the Pharmacovigilance program of India, which is uploading all its report into the WHO database, which is the VG base, and you have the VG Lives in which you can do signal detection. Within a fraction of a few seconds, you have these automated outcomes available for the disproportionality analysis tools. But please remember, qualitative review must be done additionally. It can never work in silos, quantitative metrics. These are merely numbers, and therefore you need to have it done along with the qualitative review. And another most important point regarding quantitative review outcomes is that it must be interpreted with caution. So you must be, because there are chances of spurious things coming up. Spurious associations, statistical associations can come up, and therefore you must interpret it with great caution. Next slide, please. So how do you do signal detection? So I thought of how I think of it and how we do it is to look at 4 important questions as to where to look up. Next point, please. Nithya, next, please.
Just a minute, Suparna. I think it's 1 second. It's hung, I think. Just give me a minute.
Where to look at, what to look at, and how to analyze and how to interpret. So these are four important questions. That is, where to look at, what to look at, how to analyze, and how to interpret. So if you logically think that if you can answer, or if you have laid down your methodology and you're able to do these, I think half of your job is done. So but let's first address the first question, where to look at. So where to look at, you can look at databases. These databases can either be drug safety database. One of the important databases we essentially mine for the purpose of quantitative review is the spontaneous reporting system database. And VGBase is an important database, spontaneous reporting database of the WHO. You can also look at the FDA database for spontaneous reports, that is the FAERS of the USA. Then a vaccine-associated adverse reaction reporting system of the USA, which is mainly for the vaccine, which is the VAERS. Now, you can also, these VAERS, these are vaccine surveillance databases, or the Vigilance you have for the European countries. But nowadays, people are also looking at other records, other data. For example, it is coming up in a big way that if you're mining electronic health records, then medical insurance claim. Then in the UK, they look at the general practitioners database. That is, the general practitioners, especially in COVID-19, this has been extensively used. That is, people are getting vaccinated, and these records of being vaccinated and subsequent events, medical events, which are occurring when they're coming to their designated GP or the general practitioner, and their database has been mined. So you need not necessarily look at drug safety databases. Similarly, companies or manufacturing authorization holders, they have their own database of these. So you can mine, but you have to decide as to what database or what multiple databases which you are going to look at. Similarly, where else to look at? For vaccine pharmacovigilance, published literature case reports, series, surveillance reports, important, mainly epidemiological and observational studies, and even intervention study reports also need to be looked at, because we found that large number of safety signals which have been generated in vaccines have essentially come up from observational studies or from literature, like publication of case series and case reports. Therefore, it no way undermines that quantitative measures are the only metrics for signal detection. And lastly, it is very important that we need to look at communications or recommendations made by a regulatory authority or drug safety organization reports of other countries. Let me highlight to you the important communication, say, for example, which comes up by the PRAC, that is the Pharmacovigilance Risk Assessment Committee of the European Medical Agency, the EMA. Or you look at the WHO communications with regard to safety signals. Or you look at the UK's MHRA, Medicines, Healthcare and Health Products Regulatory Agency. So you have to look up not only at one source, but at multiple sources in order to make yourself empowered with the concurrent of the safety literature of the issues which are being published, as well as if you want to do data mining or quantitative review in the database. Next slide, please.
Suparna, I'm just moving it. This seems to it's getting stuck. Yeah, go ahead, please. Yeah.
So now is what to look at. You essentially, for quantitative review, you need to look at, you have to look at the search strategies, whether you want to do a general search strategy or a customized strategy. This people say, oh, it is automated, it will do at its own. No. What is automation? What you want, you tell the computer or the software people, they're going to code it accordingly. But the mind must know, that is, what do I want? So for the purpose of searching, you must have to select appropriate ADR terms. You see, in majority of the automated tools for quantitative review, we have these MEDRA codes, that is the Medical Dictionary for Regulatory Activities. The ADRs are essentially terminology. These are hierarchical systems in which they are coded, and you have to select the codes. But it is extremely important that you select the codes appropriately. Say, for example, patients with Bell's palsy. If there are in the report, there may be people who may have reported it as facial muscle weakness, facial paralysis. Some may have come to the diagnosis of it's Bell's palsy. So you must realize that when you do a quantitative analysis, especially by the disproportionality analysis methods, you can do it only for one preferred term. But there are systems where you can, which is coming up in a big way, which is known as the cluster algorithms, where terms which are clinically coherent, they can be grouped together and you do the data mining. So it is important that we code the adverse events in a way which helps in data mining. And when you're data mining or doing a search, you have to select appropriate terms. Otherwise, it might lead to missed signals, or you might get inappropriately high statistical scores if you're not selecting appropriate terms. And you may not get adequate number of reports also, though they may be clinically similar. The other thing is that drug names also need to be coded appropriately. Say, for example, if you select, if the reports only do not state which COVID vaccine, give you an example, if you just select the active pharmaceutical ingredient as COVID vaccine, then we may not necessarily, we may get a lot amount of information, but we may not be able to selectively find out whether a particular event is occurring with one platform or not. Let me give you an example. The incidences of myocarditis and pericarditis has only been observed with mainly mRNA vaccines. It has not been observed with the adenovirus vector vaccines. So it is important that when you are doing a search strategy, you have to select appropriate drug names depending upon what you really want to look at. Then you need to look at the number of reports which you have in the database, whether it is a large database or it is a small database on which you are working at. Then for each individual report, you need to look at the completeness of information, because in an SRS, the spontaneous reporting system database, the quality of information is extremely heterogeneous. When it is healthcare providers filling in the reports, often behind, the quality of information is very good. But in several countries, including ours, persons themselves, the community, the patients, they can themselves report. In that case, the quality of information may not be so good. Even reports which we get from manufacturers, these are also incomplete. So you have to look whether there is completeness of important information. The next is the type of reporter. That is, you will often see that the pattern of reporting is often influenced by the nature of who is reporting, whether these reports are coming from the consumers or from the healthcare providers. Then the number of countries which are reporting it, because these have got an important issue or an impact on the signals which may be generated, and you need to look at whether these are country-specific issues or these are global issues. And of course, you need to look up at the statistical metrics and their trends. So it is not a one-point assessment. You have to follow the trend. Is it going up or is it going down? So these things are the ones which you need to look at. So the quantitative approach, next slide, please. So the quantitative approach to signal detection essentially entails data preparation. You must lay down the methodology, and of course, then finally, you have to do the data analysis. Next, please. Now, how to analyze, the next question is how to analyze and interpret the measures. The analysis essentially entails data mining. Data mining is, to explain to you, simply means that you're trying to systematically examine the reported adverse events using mathematical tools. And this data mining is mainly done on a database. And the outcomes, these come up as statistical values and scores. And the scores, what do they indicate? They indicate the strength of statistical association. And this essentially helps the safety evaluators to identify the safety issues that merit further in-depth analysis. So this only assists, because when you're looking at a large database, you can't hunt it out manually. So you need some technology to assist you to bring it up to your table for further analysis and review. Next slide, please. So one of the important measures of a quantitative analysis is known as the disproportionality analysis. I have willfully put this as this. So this is a prefix to the word proportionality, which means anything which is out of proportion. It may be either, in case of pharmacovigilance safety signal detection, this is mainly if you see that the combination of a vaccine in this case and an adverse event which is present in a database, whether the numbers which you observe is more than what you expect. So whether this combination of the vaccine adverse event combination, the observed count is more than the expected. How much more? That you have to set some thresholds. This threshold varies depending upon the metrics which you are using it, its clinical significance, and other data scientists often give us inputs as to what should be the optimum threshold. So, but an important point here which I wish to reiterate is that this association is statistical. And the caveat to it is that it may not necessarily be a causal association. So this is a very important point, that even if you find that the observed reporting rates are more than the expected reporting rates, it may not always be causal association, and there may be the impact of various confounders and other bias creeping in. So remember that it is not necessarily always a causal association. Next slide, please. Next slide, please. Yeah. Now, the first picture. Yeah. You go back. Yeah, just. So let me bring in an analogy to you, trying to explain, because everything is about observed and expected counts. What do you see? You see a large field, and you can see about 2 or 3 cows in this large field. So this is a big field. And in this field is large, and you know that these 3 cows which you observe, do you expect it? Yes. For this, in this large field, 3 cows, the observed is 3 cows, and you do expect 3 cows in such a large field. In fact, it can accommodate more. The next, please. Here you see about 2 cows in the highway, or if it is 3 cows also in a highway. This is your observed number of cows on the highway. Now, do you expect three cows on the highway? Now, in this case, the observed count, and we generally don't expect cows on the highway. So in this now tells you from this picture that you don't expect so many cows in this highway. So here the number of 3 of expected is far, the observed 3 cows on the highway is far more than what you expect. So this is the analogy, but the number of cows is the same. So it is important if you consider it in the context of pharmacovigilance, 3 reports in a database, whether this observed 3 reports is far more than the expected or not will essentially depend on the database and the context. So it is extremely contextual. The expected numbers have to be thought of when you analyze it with the observed rates. So the context is important when you want to say that the observed is far more than the expected. Next slide, please. So this is a where you see that the observed numbers are far more on the, and they have even led to traffic. They have halted the traffic on the road, and you can, in this case, you can see the observed numbers are far more than what you would expect, and therefore this is the analogy which we are going to use subsequently in the data. Next slide, please. So for the DPA analysis, there are basically 2 approaches. That is the traditional approach of the frequentist measures, and this is very easy to compute. And these are the 2 metrics mainly which we use: the proportional reporting ratio and the reporting odds ratio. And this is easy to compute, as you all know. But one disadvantage of this is that when you have your database is small and when your observed counts are very small, then what happens is that there is a greater chance of spurious association of false positives. And in this, you can't do much of a stratified analysis for temporal trends. It's very difficult for you to look at the effects of confounders. So therefore, nowadays, what majority of the regulatory authorities essentially look at is a Bayesian approach. Because of limited time, I'm unable to speak in details about the Bayesian approach, but a couple of things about Bayesian approach is that it doesn't only look at the current data, it looks at the posterior probability of events which are. occurred in the database. So this important lookup at the posterior distribution, the posterior probability of an event occurring in the database essentially helps you to shrink, to bring in a shrinkage. That means the credibility interval is shrinked towards the mean, as a result of which the spurious associations or the false associations can be minimized. And this is all the more effective when you have small number of observed counts. And another advantage of using this Bayesian metrics is that it allows, you can do stratification in terms of age, stratification in terms of gender, stratification in terms of time, looking at the time trends, stratification in terms of the countries or the regions from which these are reported. And the two important outcomes of this Bayesian approach, this proportionality analysis, is the IC, that is the information component used by the WHO, and the EBGM, which is used by the US people. And these have been said that these are much more stable estimates than the frequentist approach. Next slide, please. So I'll show you a hypothetical example. You know, see, people, the students and others who often look at the formula. So instead of looking at the formula, if you look, I've tried to explain how these formulas are essentially have come up. So if you have this 2x2 contingency table of a hypothetical example of the people who have got, see, there are 10 reports of vaccine A adverse event reports. Of them, there are 3 with, say, for example, GB syndrome, and the other 7 with that vaccine, you have other adverse events, but not GB syndrome. And in this entire case of this vaccine of 100 reports, you have 8 cases of GB syndrome. And there are 85 reports of other adverse events, but not with vaccine A, and these are not GB syndrome reports. These are other adverse events following immunization. So your entire database is a total of 100. And what is your observed count of the vaccine with GBS? That is, you have 3 reports. And what are the total GBS reports? So you have 8 reports. That means if you look at what is the frequency of GBS syndrome in the entire database, it is 8 out of 100. So that is 8%. So that is, but what is your observed count? Your observed count is 3. Now you need to compute your expected count. So if the simplest approach of computing the expected count is, it will be 8% of the total number of reports which you have with the vaccine. So that is 8% of 10. Because you have 10 reports with your vaccine A. So 8% of 10 reports means it is 0.8. So the relative reporting ratio is observed by expected. So you observe 3 reports, but you expect 0.8 reports. So the observed versus expected ratio is 3.75. Now, if you get that, this formula has essentially come from this understanding as the reporting ratio, relative reporting ratio. The other important tool which we use is the proportional reporting ratio. Now, how is it different and why is it different? Remember that you have these 3 reports, but for calculating events, we do not take the reports with the drug or the vaccine in question. That means here you see that the frequency of adverse events is only 5, not 8 out of 100, but it is 5 out of 90. So you are excluding these 3 reports because these are related to the vaccine, and because the vaccine has 10 reports, you're deleting those 10 from the total count. So for proportional reporting ratio, it comes to 5 out of 90, that is 6, and therefore your expected is now 6% of 10, that is 0.6, and this ratio is your proportional reporting ratio of 5. And the reporting odds ratio is similar to the odds ratio that we computed, and that comes to 7.3. So we see that for 100 reports, vaccine reports database, your metrics vary from about 4 to 7.3. Yes, there is a lot of variability. This essentially occurs when your database is small, but you will always see whatever metrics you use, they indicate the same thing. That is, there is a positive association is coming. But what happens is that when you calculate the confidence intervals, they will have very large confidence intervals likely, and therefore there is a need when you are doing this to be substituted by what is known as the information, the Bayesian metrics, that is the information components, or the EBGM. Nithya, next slide, please. So for calculating these, as I was telling you, you need to set your thresholds. And these thresholds are mainly indicated to minimize the false positive signals. And some of the thresholds which have been set by certain regulatory authorities is given here. That is, you have a proportional reporting ratio of 2 or more than 2. And this is your confidence interval. And these are some of the values for setting thresholds. These thresholds are essentially dictated by your clinical inputs, the relevance, the public health importance, how medically important these events are, and not merely looking into the data elements of it. If you see after qualitative review that using these thresholds, the number of spurious associations in qualitative review is appearing to be more. There are effective confounders. Then what is happening is that you have certain most strict threshold in order to minimize this background noise. Next, please. So these are some of the newer approaches and tools. It is the clustering algorithms, the rapid cycle analysis or the change point analysis, the stratified disproportionality, the multivariate analysis, and instead of the data mining, now people are using text mining, natural language processing. Is that in the narratives, these information is being captured and mined. Even reference mining is now being used. So I'll speak a couple of things about these newer approaches. Next slide, please. So the newer approaches, that is the cluster algorithms, this is a new approach which is mainly being used both for vaccines as well as for drugs. They will identify and investigate the signals. And here, instead of mining a single term which we use for disproportionality analysis, you do a clustering of important clinical events. Say, for example, you have extrapyramidal symptoms for a particular drug. So you don't only mine for extrapyramidal symptoms, you can have dystonia, you can have hypertonia, you can have hypotonia. So clinically coherent things. Similarly, for Bell's palsy, not only Bell's palsy, you can have facial paralysis, you can have facial muscle weakness. So clinically coherent terms or entities are put together as a cluster to effectively help in it instead of course, you can do what is known as the standardized meta query searches also instead of looking at the only single preferred terms for single item searches. And based on that, there are certain data scientists who come up with temporal scan statistics, that is for clustering, how this cluster is moving over the time, the trends. These are essentially being used, and this tool has essentially been used for the COVID vaccines by both the European agencies as well as by the US people. And the other thing which has come up, especially during this COVID time and also with the influenza vaccine studies, these are the rapid cycle analysis. The important feature of rapid cycle analysis is that it is used or helpful for near real-time analysis. What is the meaning of this? The meaning of this is that in the conventional approaches for drugs, we do it at a time interval. That is, for example, the WHO does it at a quarterly interval. So the quarterly, the data collects and then you do the analysis. You wait. That is the traditional, it is a non-sequential analysis. You allow the data to accumulate, and at the end of the study or at the end of that period, then you analyze. But this method of rapid cycle analysis is a continuous method. That is in a real time. You do it as the data comes in. So with each data, it's the dynamic metrics. And this essentially generates a very complex measure that is known as a sequential probability rate ratio, SPRT, is being computed. And this method of rapid cycle analysis gives you the time trends, very quick time trends from day to day, from weekly, and therefore this helps for rapid safety communication. And this is all the more what we have done is important for the pandemic. Next slide, please. So this and the other newer approaches which I was telling you is that not only looking at the databases of drug safety. See, this was a very nice and very recent paper which has come on the PMJ Global, where their electronic healthcare data has been mined for safety signal detection. And here also they have used the continuous sequential rapid cycle analysis method. So that is why that you do not necessarily have to look at only the drug safety database. These are other databases which are coming up for having a look at for doing signal detection. Next, please. So the finally, you should look at the following things. That is the SNP, they say, the strength of the association, try out and find what is new, whether it is clinically important. That is very important, clinically important, both at an individual level and for vaccines more at a public health level. And what are the identifiable risk factors and how it can be prevented. Next slide, please. Now, see, these are, what are the special issues in vaccine safety signal detection? See, the question is, which spontaneous reporting database is going to be selected for mining? Will you use a vaccine-only safety database, or are you going to use a database which contains both vaccine adverse events as well as drugs? You know, the answer to this is not easy. See, various agencies, for the US FDA, they have used only the vaccine VARS database, that is the vaccine-only database. Whereas the WHO has used, the UNC uses both the VG base, which contains vaccine reports as well as drug reports. There are advantages and disadvantages of each of them. If you use a vaccine-only database, then there are chances that you might miss out certain new events which can come up, which are not associated or not previously reported with other vaccines. But often if you use both the drugs and a vaccine database, the database is large and you have to look at the expected counts are likely to be confounded by the nature, because the people who are taking the vaccines will be quite discrete from the people, entire population of the spontaneous reporting system. So these are the things which have to be very carefully thought of. Similarly, another question comes up for vaccine signal detection is, how do you code for the adverse events of special interest? See, the MEDRA codes which are used for data mining, these are not completely synchronized, and therefore efforts to map these MEDRA codes with more specific vaccine-related adverse events, that is, which we have the Brighton collaboration case definition, this mapping is essentially needed. Then similar clinical entities, but each AE is coded differently. So how do we take care of that? The answer to this is we try to club together similar clinical entities and we try to do event cluster analysis. So these are certain special issues which come up for vaccines and for vaccine safety signal detection, but are not. And the last is most important. How do we assess for the effect of confounders? You see, how do we adjust for age? So there are methods which have come out where we do adjustments for these confounded, that is weighted averages come out. Then in immunization and for vaccination, people, especially in pediatric practice, multiple vaccines are given within a short span of time. In one visit, the children get more than one vaccine. So how do you analyze such reports where you have multiple vaccines at the same time and you have an adverse event? So these are important confounders or important issues which mainly occur with vaccines, and therefore we have to adjust for each of them and find out novel tools in order to when you are going to analyze it and interpret it. So at the end of my talk, you would like to know what are we doing in India about vaccine signal detection? You know, India, you know that we have a very robust AEFI reporting and monitoring system. But the main focus of this AEFI, and Professor Arora is there, who really has been doing a marvelous job in this, and Madam Satinder Aneja, and and several of us are in the National AEFI Committee. We have a robust system for AEFI monitoring, but this is mainly focused towards childhood and maternal immunization. But till date, we do not have a functional vaccine signal detection system in India. But the ministry has already formulated, and we have Professor Rakesh Agarwal, who is the chairperson of this committee, and we are contributing and to find out, and we're having deliberations to chalk out a framework as to how we are going to do vaccine safety detection in India. And of course, our important goal is that we have to do it from a national perspective. There are several limitations of vaccine safety signal detection, especially in our setting, because our AEFI reporting is suboptimal. So the existing database, you just one more point, just press the Nithya, just press the here, I'll show some figures. You see that we have very suboptimal, sorry, the previous one. I think it didn't come up. We have suboptimal reporting. You know the huge number of COVID-19 vaccines which we have given, but the AEFI reporting rate per 1 lakh doses of COVID vaccine administered in our country is only 4.22. So we have 4.22 per 1 lakh doses of COVID vaccine administered. And in the SafeFact, which is the WHO database which we are using for uploading our AEFIs, we have from 2013 till 2020-21, I think it is about 11,000 or 12,000 reports. And these reports, the sources of this information is from the ITSU. So that is a technical unit which is looking at the adverse events following immunization. So in a country like ours, where we are, the reporting culture of AEFI is poor, and as per the recommendations of the AEFI, we only report serious and severe reactions. But when we want to have a full-fledged vaccine signal detection, we need to report even minor ones, because that helps us in getting background rates correct at the expected rates. So therefore, it is exceedingly challenging, but we will go forward and we'll find a way to best suits our needs in resource limited settings and where the reporting culture is suboptimal. Next, please. So these are the challenges.
May I request you to wrap up, please?
Yes. So these are the challenges. These are the challenges of the heterogeneity, selection of comparative groups, and adjusting for confounding factors and in sparse limiting. Last slide, please. So these are the take-home messages. That is, statistical measures should not be interpreted alone or out of context. Qualitative review cannot be substituted. There are newer tools and approaches that are emerging to minimize bias, but of course, due diligence is needed. And the last thank you slide. So thank you very much. And I'm sorry for the technical glitch which happened in sharing of my screen.
Thank you, Suparna. There are some questions that have come up in the chat box, which I will read out to you. The vaccines are always in some or other controversy. So is it due to the fact that it will be used rampantly in healthy people, or is it the perception of the people that they consider vaccines or biologics to be dangerous?
I think this is a question which essentially involves the attitude and the understanding. Of course, people have a perception that biologics have some inherent greater safety issues than small molecules, but this may not necessarily be true, because taking into considerations that thousands were receiving vaccines and how safe they are, I think this is more of a myth than a truth.
Okay, the next question is, how is vaccine detect? No, there is one here. If we want to plan to detect signal, then how we determine the numbers of reports to look at, and what factors determine the numbers required to be looked at. Second part is how researchers could have found out blood clots during the COVID-19 vaccine.
So I'll first answer the second one. How did they find out blood clots? These were essentially epidemic case reports and observational studies. These were followed up subsequently by quantitative metrics, but the initial cue was essentially published reports and observations which were seen. So these were all qualitative methods which came up, but these were substantiated subsequently by quantitative. And the first one, the question is as to how big a sample size or the database should be. I would request you to read a very recent paper by a WHO group, Ola Koster and others, where they have said at a national level, they have said how small you need to have at least a 5,000 database. They have done certain statistical measures to find out what should be an optimum age. So at a national level, at least a 5,000 database should be enough. But if you want to do more stratified age, then about 50,000 is what is recommended for you. So I think instead of my recommendation or my understanding from the literature and the little experience is that we should start off with qualitative review from observational studies and look up, and therefore thereafter be followed up by quantitative metrics, because the quantitative metrics for vaccine signal detection are in no way very different from that of small molecules. The metrics are the same, but the threshold which you set and adjust it for confounders, that is where your due diligence is needed.
Thank you very much for this comment about qualitative evaluation of the data. And I really want to highlight how important this is, because our hopefully our audience contains AMC coordinators. And it's so important that you send us good quality narratives, because otherwise we are looking at secondary data, and when we're doing signal review, if the narratives are poor, of poor quality, we cannot understand what you're trying to say and whether there is an association and whether that we should even consider this as a signal. And I think this is a very important thing that you have said. Anyway, the next question is, how is vaccine signal detection from routine signal detection?
I think I have already explained to you the small differences of vaccine. Principles are the same, but critically reviewing it, analyzing it, and adjusting for confounders is important. And the rapidity with which you do it and the dynamic metrics is what here, because for vaccine signal detection, this is a public health issue. So the promptness and the newer tools is what distinguishes it from the conventional molecule. Otherwise, in principle, it is the same.
Thank you, Suparna. This was a very lovely talk and very nice discussion at the end. Thank you for the questions, also for the participants. I think I will hand over now to Dr. Agarwal to continue with the session. Dr. Agarwal?
Yeah, thank you. I don't know whether you can see me, but that's fine. Good morning. So our next speaker is Dr. Sunita Shukla. Dr. Shukla is an ENT surgeon and also has a diploma in hospital administration. Her primary area of work is allergy and asthma, in which she has received training at CMC Vellore, as well as abroad in University of Colorado and at Mass General Hospital in Boston. Dr. Sunita has established a separate organization, Mumbai Allergy Center, and she heads that center, which also offers vaccine allergy testing. And she's also set up this testing at KEMH Hospital. Dr. Shukla, please.
Good afternoon, everyone. Thank you, sir, for the kind introduction. I thank Dr. Nithya ma'am for giving me this opportunity, where we can talk about the real world evidence in drug and vaccine allergy and what we are doing. Is my slide visible?
Yes, Sunita, very clear.
Yes, Sunita, yes.
Thank you. So today we'll be talking about allergy, what we can see clinically as a physician. So drug allergy is an immunological mediated response to any pharmaceutical or formulation agent in an already sensitized person. So most of the time, these drug reactions can be a type A reaction, which is a dose-dependent, predictable, more common reaction seen in more than 80% of the people. And these are usually the overdose, side effects, and the drug interactions. The type B reaction, which is dose-independent, unpredictable, and less common, are seen in 20%, and these are various types of intolerance, and most important is a drug allergy. So today we'll be talking about the most commonest, the immunological reaction, the type 1 reaction, which we see in drug allergy, where the clinical symptoms are because of the IgE mediated type, which is anaphylaxis, urticaria, angioedema, bronchospasm, hypotension, and so on. So the mechanism of any IgE mediated reaction is that the body which is already sensitized has this IgE antibody to that specific allergen. So these IgE antibodies, they are present on the mast cells, and there are many which are freely circulating in the blood. But when an allergen comes in contact with these IgE antibodies, it causes deactivation of the mast cells, and in this process there is degranulation of the mast cells, which releases many chemical mediators like histamine, cytokines, and so on. which gives the typical symptoms of these allergies, which can be bronchospasm, edema, hypotension, and so on. So drug allergy diagnosis itself is a enigma, because most of the time these patients are admitted in the hospital, they are on multiple drugs, and which drug is causing the symptoms, we usually don't know how to diagnose it. So for any drug allergy, for diagnosis, one of the most important thing is a history taking. Here the onset of the symptoms and the temporal relation of giving the drug is very important. In examination, these could be the local signs and symptoms as well as the systemic symptoms, including the vital parameters. We can do some in vivo tests, which are very important in drug allergies. These are the skin prick testing, intradermal testing, and an oral or an IV challenge. There are some in vitro tests, the blood test, which can be routinely done, which are the drug-specific IgE, basophil activation test, and the serum tryptase level. But these tests are usually not available everywhere. In Mumbai, only serum tryptase is done by one lab only, and the BAT is still not established here. So skin prick test is a gold standard diagnostic method for any IgE mediated allergies. The reason being, it is a very simple test which can be done once a person is trained for it, and it tells you the significance of the IgE which are present on the mast cells. This is a small video clip where I'm showing the skin prick test. Here we can see the drop of allergen is placed on the forearm, and we do a micro prick with the needle. And when this is done, the allergen is introduced below the stratum corneum, and this activates the degranulation of the mast cells if specific IgE antibodies are present. And this we see clinically as a wheal and flare reaction within 15 to 20 minutes. The best part about this test is it can be easily done and the patient can see the reaction themselves. But in drug allergy, only SPT is not adequate. We do something which is called as intradermal test, where a small amount of diluted drug is injected into the intradermal layer. And again, the reaction which we see is a wheal and flare reaction in 15 to 20 minutes. Drug challenge test is done once the skin prick test and the intradermal test is over and they are negative. And this is the gold standard for diagnostics drug allergy. It can be done via the oral or the IV route. And it is a multiple step approach where the diluted form of the drug is introduced in the body and we go on to the full concentration. And this is done at a regular interval with monitoring of the important vital parameters. And we see whether the patient tolerates it or not. But this should be done only at a center which has adequate infrastructure to manage any anaphylaxis or reaction. So there are some few interesting cases I will like to show. Like this is a case of Ciplox allergy. A young male with a childhood history of allergic rhinitis, food allergy. He reacted to Norflox in 2009 and then to amoxiclav in 2019. Now this patient was referred to us because he was supposed to undergo an elective dental procedure, and the dentist said that we will be giving you Ciplox. Because the patient was educated and aware, he said that no, I don't tolerate the drugs very well, so I want to rule out allergy to it before I take the drug. So when we did a drug allergy, as we can see here, the initial pictures are of the skin prick test, and as we go down, these are the intradermal tests, and here with the full concentration, that is 1:1, we can see a huge wheal and a flare reaction, which is more than 2 centimeter and 4 centimeter. Now, when we injected this, the patient immediately started getting rashes on the ears and the trunk. Now, if this patient takes even 1 dose of Ciplox, chances of him having anaphylaxis is very high. So in such patients, we label them as a drug allergy and we ask them to avoid these drugs. So the next question was, if he is allergic to this, what other antibiotics can we give? So with discussion with the dentist, we decided to go for cephalosporin and we tested him for ceftriaxone. And here we can see the skin prick test and the intradermal test, both were negative. So we went ahead with the oral challenge test with cefixime, which the patient tolerated very well, and he underwent the dental procedure with the cephalosporin coverage. Coming to quinolones, this is a young doctor who is only asthmatic, which is non-allergic, and she has a family history of allergic rhinitis. But in 2019, she took a drug, one single dose of levoflox, and this is a typical fixed drug eruption which happens on the foot, and this is how it healed within 2 to 3 weeks. Now, 1 year later, she took Ciplox TZ for her diarrhea, but within an hour of taking Ciplox, she got the same rash at the same spot, and this is how it healed within a 1 month by desquamation and hyperpigmentation. So this is very typical of fixed drug eruption, that whenever you take the offending drug, the reactions happen at the same spot. And this is important because if we have to test such people, we have to do a local drug testing on these spots only, because all the mast cells and the IgE are concentrated at this location. These are interesting photos of a FDE for doxycycline. Here we can see how this hyperpigmentation happens, and that is how we diagnose it. These are very severe reaction to single dosage of ofloxacin, amoxiclav, and this is a Stevens-Johnson syndrome, which could be life-threatening also. Another interesting case is of a doctor who had lignocaine allergy. She was labeled this 20 years back when she was undergoing a cataract surgery. Now she had to undergo a root canal treatment, and she was because very fearful for this lignocaine allergy, she underwent this RCT process without a lignocaine. We can imagine how painful it must have been. But now she had to undergo another procedure, and she was referred to me. When we did a skin prick test, here you can see after doing a SPT, then we went ahead with the serial dilutions and then the concentrated form. Everywhere we didn't find any reaction. So after doing the 1% concentration, we did the 2% and everything was negative. Then we went to the intramuscular challenge test where we injected increased quantity in the subcutaneous space, and she tolerated it very well. Now this lady has again done her RCT but under lignocaine. So this is what happens when we mislabel a patient, and this is 20 years back and she was still having the label unless we test it and we can delabel it. This is another case of an old lady who was awaiting some surgery, and she was referred to rule out allergy to cephalosporin. And here we can see that when the SPT was negative, but as we were increasing the dilution with 1:1, we got a very severe wheal and flare reaction. Here the dilutions and concentration very important, because every drug has a different protocol, because there is something called as a non-irritating concentration, and we have protocols for each and every one, and that is how we decide how much to dilute and which is the maximum concentration where we can go in intradermal testing. Next case is of a male who presented with the recurrent episode of this angioedema. He had comorbidities like diabetes, hypertension, sleep apnea. But when we check, there are multiple reasons for such angioedema, including one important thing is drugs. But we have to rule out all the other causes like allergies, hereditary angioedema, inflammatory conditions in the body, and everything was negative. So after taking a detailed history, we realized that it could be a drug-induced angioedema, because the patient was recently started on aspirin and olmesartan. So when we discontinued these two drugs, within 48 hours, the edema subsided. This patient was in the ICU twice in a period of 2 months because of this drug. So many times we don't realize, but these angioedemas can be because of the drugs also. This is a very interesting case of a young ICU sister in charge. She was admitted for COVID in the hospital. She did had a family history of allergies and atopy, and she developed some rashes, for which she was given an injection Avil, and she collapsed. When she was referred to us, we did her Avil drug testing, and here we can see that even with 1:10 concentration of intradermal testing, we could see a huge wheal and a flare reaction. Now, in such case, whenever we get a reaction, then we don't go for the second testing, because it could be a catastrophe. And this is a very rare event. We could find only 3 such cases on Google search for Avil injection, which actually is a drug which is used for so many allergies. Another case of a lady who presented with multiple drug allergies. She had come to us for COVID vaccine testing, and on taking the history, we realized that she has allergies to cetirizine. But though it is so rare, we decided to test it, and we did an oral challenge test for her because cetirizine here is not available in the injectable form. And with 1/10 of the dose, she started developing rashes and intense itching. So whenever a patient gets any reaction, we don't go further with a further concentration and stop only. So this is a setting which is also a rare case. Why one should refer to an allergy specialist? The most important thing is mislabeling. The example which I gave was the lignocaine allergy, and this most of the time the patient will carry the label lifelong. Many of the times a patient with this label is given a lot of antibiotics, which leads to antibiotic resistance and increase the usage of other antibiotics, which can be more toxic and most expensive. So, and one more important thing is that we should identify the true allergic patient, and then we can classify them in the mild, moderate, or the severe risk. This is important so that we can educate the patient about what allergies they have, and if any reaction happens, if they are away from a hospital, what necessary step they should take so that they reach the hospital safely. The treatment for any drug allergy is avoidance of that culprit drug, and also the family of the same drug, because most of these are cross-reactive, and we have a cross-reactivity chart. Depending on which drug the patient is allergic to, we decide what drug is safe and whether we should give them or not. The other treatment is desensitization, because many times we don't have any other options of giving any other drug. This is true for drugs like aspirin or the anti-cancer drug, where we have to give that drug. In such case scenario, we can desensitize the patient, where we are giving the drug in very small quantities in an ICU setup, and we gradually increase it. Many times we do pre-medicate the patient also before this. So my talk will be not complete if in this COVID era I don't talk about the COVID vaccine allergy. Any vaccine allergy can present in two forms. First, when we inject, it could be a large local reaction like this, or it could be a systemic reaction, which could be anaphylaxis in any form, mild, moderate, severe, and it could have urticaria, rashes, bronchospasm, and so on. This is a picture which shows how vaccine-induced allergic reaction happens. But to simplify this, I made this diagram where you can see that the center is the mast cells. Most of the time, these reactions could be either IgE-mediated reactions, or there could be many other factors like multiple, the complement system, the tryptase, the IL system, and a direct activation also. So any of these factors leads to the degranulation of the mast cells. It releases a lot of chemicals like histamine, cytokines, interleukins, and this actually gives a allergic reaction. So initially, when people used to think that vaccine allergy could be IgE mediated, but now it is proven that there are so many other causes, and that's why it becomes very difficult to predict which patient will have a reaction to the vaccine. So to the vaccine, the reaction which is due to the vaccine can be because of two things. One is the vaccine component, the biological component itself, or the excipients which are present in the vaccine to give them more stability and for preservation. So these excipients normally are egg proteins, formaldehyde, gelatin. But now with the COVID vaccine, it is PEG, that is polyethylene glycol, and polysorbate. The COVID vaccine allergen, as I said, in most of the mRNA vaccines, it is PEG, and in the adenovirus vector vaccine, it is polysorbate 80. Now, these products, whether they cause the IgE-mediated mast cell degranulation or some other non-IgE-mediated mechanism, is what I showed in the previous slides. But as this polysorbate and PEG is so common in so many of the medicinal products, like if I have shown this chart of polysorbate 80, so many vaccines like the influenza, hepatitis, DTaP, all these things have polysorbate 80 as one of the excipients. Also, PEG is so common nowadays. We find it in so many products like the steroid injections and the depo-provera and so on. So these excipients are very common, but most of the time these don't give a reaction because all of us are exposed to this component somewhere or else in our lifetime. Like very common example is that we have this PEG or polysorbitity in our toothpaste also, in cosmetic product also. So we all are actually sensitized to it already. But the government guidelines which came up in January 21 for the COVID vaccine was very clear that a patient if has an anaphylaxis to a previous dose of COVID-19 should not be given, or if there is any reaction to a vaccine or injectable therapies or a drug or a food item should not be given these vaccines. Unfortunately, the criteria was very clear that it has to be an anaphylaxis. But this was misinterpreted, and what better example to give than this? This is a young boy who was mislabeled as multiple drug allergies by a GP. And this, when he went to the Kasturba hospital, which is the only ID hospital in Mumbai, they refused to give him the vaccine, saying that he is allergic to so many drugs. And he was refused. So coming to the contraindications and precautions, this is the World Allergy Organization chart, which was released in Feb 21. Here, the green is when we can vaccinate, yellow is with precaution, and red is contraindicated. You can see here, if a patient has any history of inhalation allergy, drug allergies, we can go ahead with the vaccination with a 15 to 30 minutes of observation. However, if a patient had any anaphylaxis before, or if it is an idiopathic anaphylaxis, still we can give a vaccine to this patient, provided the patient is observed, and if there is any doubt, better to refer to an allergist. But however, the most important was the contraindication, which happened for only the anaphylaxis to a COVID vaccine before. Otherwise, this patient can be easily vaccinated. But however, is my slide seen?
Slides are seen.
Yes, Sunita, you have moved from the slideshow to the usual format. Yeah, now again slideshow. Please go ahead.
So this was a contraindicated only for a patient who had an anaphylaxis to the vaccine before, or to any of the component like PEG. But however, these patients should be referred to an allergy specialist where they can do a detailed testing and decide whether the patient should receive vaccination or not. Coming to a final word which was recently released by various allergy organizations like the World Allergy Organization, the American Association, the British Society, Australian, Philippines. This is a gist of that decision, how people treat them now. If there is a history of PEG or polysorbate anaphylaxis of severe reaction, then there is a high risk of getting any reaction to the vaccine, and such patients should be referred for skin testing. However, if it is not so severe and there is a reaction to an injectable medicine, a prior vaccine, or any other allergen, this patient are at medium risk and they can undergo routine vaccination for 30-minute observation. And with any other allergies, whatever history patient can have, it is a very low risk, and the patient can undergo a vaccination with 15 minutes of observation. So this was a data collection from Mumbai Allergy Center, which we published recently in JAPI, where we evaluated the allergic reaction following COVID-19 vaccination in patients who had documented allergies before. So we had 227 patients in this, where we can see they had so many allergies, including drug allergy, food allergy, anaphylaxis, allergic rhinitis, asthma, and so on. And we collected the data whether they have taken the COVID vaccine, which vaccine they had taken, whether it is the first dose or the second dose. And as we can see that none of our patients had any severe reaction to the vaccine. Only 3 patients had mild itching, which was subsided with taking an antihistaminic. But this doesn't come into the severe reactions of the COVID vaccine. So this is the COVID vaccine component testing which can be done for the excipients. Here we can see the vaccine skin testing and the various other allergens. And at our center, we follow this Harvard protocol where if the patient wants to do a COVID vaccine component test, we actually test for PEG and polysorbate 80. This protocol was for last year when we started this in June. And why we need to do it for PEG, though we don't have an mRNA vaccine in India. We have Covishield, which has polysorbate 80. But most of the time, basically this PEG and polysorbate 80 are cross-reactive. And because PEG is so commonly used in so many medicinal products, it was advised that better to do testing for both of them, because if a patient has any cross-reactivity, we can easily detect it. This is how it was done. The initial is the skin prick test, and then we can see the three other intradermal testing is for PEG, and the last is for P80, full concentration. And if the patient didn't have any reaction, we ask the patient to go for vaccine testing. So in our center, we had 27 patients whom we tested for this hypersensitivity to PEG and polysorbate 80, and all of them were negative. I didn't find a single patient at my center, and all these patients then successfully overwent for their COVID vaccine test with either one or the second dose. And none of them had any reaction after that. This is a prospective study which we are doing at KEM now, and here we are doing skin testing in patients who are having any allergies, various allergies or a vaccine allergy. Here we test only for polysorbate 80, where we can dilute it from 1:100 to the maximum concentration. If there is no reaction, we do the COVID vaccine testing with Covishield, and again this starts from a dilution and to the full concentration. This patient did not show any reaction to any of the vaccine, and then we advise him to take the vaccine without any issues. But we can see in this patient, with the full concentration of the Covishield vaccine, we got a wheal and flare reaction, which was the wheal was 8 millimeter and the flare was of 15 millimeter. Now, this patient had already taken first dose of the COVID vaccine, and after taking it, he had some rashes in the arm. That's all, nothing else. So we, in such a scenario, we then ask the patient to take the vaccine under our observation. There is another way of giving it. We can go for the slow graded vaccination, where part of the vaccine is given, and if the patient tolerates it, then we go for the full dose after observing. So this is what we are doing at KEM now. So to conclude, no patient with food or drug allergies developed severe allergic reaction post-vaccination in our data collection. The vaccination guidelines should be revised for the food and drug allergies, and they should include only patient which has severe allergic reaction to PEG, P80, the injectables, vaccine or allergen. And people should not misinterpret this statement that if it is anaphylaxis, then only you ask the patient not to take it, or better, the patient can be referred to an allergy specialist. We have this facility at KEM hospital. After doing a assessment, doing a skin testing, we can decide whether the patient should be given the vaccine either under observation or with a slow graded manner. There is no doubt about it. It is a well-known fact now that the benefits of COVID vaccination far outweighs the risk of allergic reaction, and we need this to be implemented so that the vaccination against COVID-19 is the most important for defeating this pandemic. I thank you all for your patient listening.
Thanks, Sunita. If there are any questions, we can take those up. But I would say word over, nobody has gone on to allergy testing for COVID vaccines. Just because we can do it doesn't mean that we really need to do it. The initial fear that was there, even of anaphylaxis, wasn't borne out when one went to looks at the real life experience. In the trial, some cases were seen, but subsequently that was not held to be true. And I don't think as of today, we should really be recommending allergy testing for COVID vaccines for such people.
Yes, sir. you rightly said we don't need to for majority of the patient unless, as we said, there is anaphylaxis to the vaccine itself or to one of the component. But this statement was misinterpreted, and as we can see, there are so many of the allergy patients who are so fearful. It's a myth.
I totally agree. In our country, a question was asked about steroids. It was not that the indications were very clear, but people received steroids even when they did not have severe disease. There is something that needs to be done to our medical professionals. The fear of God or whatever law needs to be put into them, because they just don't read very carefully. They look at things very non-specifically, isn't it?
Yes, sir.
In the chat box, there are some questions.
Rakesh, if I can make a comment here based on the...
Sure, sir. Sure, sir.
So, thank you very much, Sunita. Very excellent talk. And in fact, you have given some new dimensions also. But just going back from the National AEFI database, one of the key concerns were that of this allergy and anaphylaxis. And that's why half an hour waiting time was given. And it is, as all of us are aware, that it is only in September, October that two programs came up in back to back. One was near home COVID vaccination centers, and then Har Ghar Dastak program, which came up in November and which is going on. And this confidence came primarily when 1 billion doses were already given, and severe allergy was extremely uncommon. Allergic reactions are about 1 in 15,000 to 20,000 from the national database, AEFI database, I'm saying, because that is reasonably good, because allergies will occur within first 72 hours, means within few hours only. So that is quite a valid data. And I think only 2 or 3 individuals lost their lives because of anaphylaxis. So overall, what Rakesh is saying, completely agree that these vaccines have been very safe, and allergic reactions did not really pose a national program. And we know 90% was Covishield and 10% was Covaxin. Thank you very much.
Yes, sir. So as rightly said, these anaphylaxis world over, the studies showed that if any anaphylaxis happened, it will be within the first 17 minutes. That was the time calculated. So our guidelines were very good when we had an observation period of 30 minutes because it included everything. But now we know, as we are becoming wise and we know COVID and what all the vaccination is doing, as initially we never knew what is causing it, but now we have literature where it's not only IG mediated, there's so many other reasons which we can't predict. The skin testing is only for the IgE one, but luckily because we could do it and we could convince the patient, at least those 27 patients have taken the vaccines. There are so many who are the age is more, they have comorbidities, and in such patient vaccination becomes so important. So if we can help them prove that, okay, chances of reactions are less and you should go ahead with observation, I think it's a good say, so that we defeat this pandemic together.
Any questions anybody has? Because in the chat box I did look for, but basically those questions are those that you already addressed. How do we test for allergies to vaccines? Nithya, any other questions? Otherwise, we'll close the session.
Yes, please.
Yeah, I think you should close the session because I don't see any new questions.
So thanks, Dr. Suparna, and thanks, and Dr. Shukla. With that, we close this session. Thank you.
Thank you, sir.
Thank you also, Nithya. And we'll go ahead to the next session.
Thank you to both chairpersons and speakers. Thank you so much. Yeah. Shruti, over to you.
Thank you. This session has been helped in enriching our knowledge during safety of the vaccines and vaccine allergies. I would like to express my sincere gratitude to Dr. Sunita Shukla and the chairpersons, Dr. Urmila Thatre Madam and Rakesh Agarwal sir, for such a wonderful session. Now I would like to hand over the proceedings to my colleague, Dr. Sukant.
Sukant, please get started.
So thank you, Dr. Shruti. Moving forward to the final session of the workshop, I would like to introduce the chairperson for the final session, and that is Dr. N.K. Arora, sir, and Dr. Usha Rani, ma'am, who would be chairing the final session. Dr. N.K. Arora, sir, is a pediatric gastroenterologist by training and has been trained in epidemiology and biostatistics from Australia. He's currently the executive director of the Incline Trust International. He's extensively involved in policy making, program planning, and implementation. He's also the chairman of India's COVID-19 working group of the National Technical Advisory Group on Immunization (NTAGI), and is also the president of AIIMS Deoghar. We welcome you, sir. Dr. Usha Rani ma'am is currently the professor and head of the Department of Clinical Pharmacology in Nizam's Institute of Medical Sciences, Hyderabad. Ma'am is a close friend of our department, and she has helped us in setting up a lot of pharmacodynamic practicals for DM curriculum over the years. She is the chairperson of the Data Safety Monitoring Board and a member secretary of the Ethics Committee for Stem Cell Research at NIMS Hyderabad. She is also the member of DBT Task Force on translational research on development of products and processes from the medicinal plants. We welcome Dr. N.K. Arora sir and Dr. Usha Rani ma'am and request them to kindly introduce the speakers for the final session and begin the proceedings. I would request the IT team to make the speakers as co-host.
Sure. Would you like to begin, or should I say a few words?
You can start, sir.
Yeah. So good afternoon, friends, and thank you very much, organizers, for involving in this very important. I have attended the previous session, and I find it extremely useful and timely and relevant. So in this session, we are basically going to talk about issues related to risk and benefit. And I'm sure the panel we are going to have in this is very distinguished, and particularly Vivek Ahuja. I know him right from the residency days. In fact, we have grown together on many for last, I think, almost 30 years. So I thought that the panelists would be clearly addressing the issues of risk and benefit and help us understand what are these, how do we quantify. But an important component of assessing risk and benefit is that we need to understand and take it in the context for which we are talking about risk and benefit. So whether it is cultural, social context, economic context, epidemiology is very important. So for example, in India, natural infection with COVID is so important, so rampant or widespread. So there the risk and benefit of various interventions. And finally, I also, I'm sure people would agree that existing health infrastructure and access would also make the context important. So with this background, I have great pleasure in inviting Dr. Vivek Ahuja, who has done his MD Pharmacology from AIIMS and subsequently he's done his business administration. He is currently Vice President, Medical Affairs and Global Head Global Pharmacovigilance at Sun Pharma. Another very important point is that throughout I know that he is very passionate about pharmacovigilance as a science, and throughout his post-MD career, most of the time he has been involved in pharmacovigilance, compliance, operations, training, monitoring, regulatory issues related to pharmacovigilance. So over to Vivek, and we would look forward to hear you about this automation and artificial intelligence. Vivek.
Thank you very much, sir. Dr. Nithya, could you please confirm if you can hear me well and see my screen?
All good, Vivek.
Okay, thank you very much, sir. It's an honor and privilege to be presenting in front of you, and thank you for the very kind introduction. So I'll be focusing on the technology aspects with respect to how they can play an important role in the benefit risk evaluation. So just a disclaimer slide. The views expressed here are my individual views. So what you're seeing on the left is a Ford Model T. It is colloquially known as the Tin Lizzie, and this was an automobile produced by Ford Motor Company in 1908. And automobiles were produced from 1880, and they were mostly scarce, expensive, and often unreliable. What you're seeing on the left is Model S of Tesla. With the longest range and quickest acceleration of any electric vehicle in production, Model S Plaid is the highest performing sedan ever built. Top speed 200 miles an hour, range of driving 396 miles, 1020 horsepower, 1.99 seconds from 0 to 60. It took us 100 years to make this progress. Let's see how much progress we have made in the last 15 years in the field of pharmacovigilance. These are some issues that we've been tracking over the past years. And I was just thinking to myself, how have these issues shaped over the past 15 years? Underreporting was always an issue, and it's no longer that critical an issue worldwide. If I talk about it, of course, in certain developing countries, it is still an issue, but pretty much we have moved on and we are addressing more technical issues than merely addressing the issues of due diligence of how to collect data. Harmonization of regulations, more countries, more drugs, more populations, more regulations, and very little harmonization. In fact, we have scattered a lot, but at the same time, there are certain good areas. For example, the European Union regulations in pharmacovigilance have been harmonized, the Gulf regulations have been harmonized, Eurasian regulations have been harmonized, but by and large, they are still scattered. Technology. Now, technology has progressed, but there is still not fully harnessed to its full potential. So there are people who know pharmacovigilance and want to learn technology, and then there are people who know technology and want to learn pharmacovigilance. That's how this field is behaving right now. And lastly, special PV requirements for new age medicines. So with personalized medicine, stem cell therapy, gene editing, all these areas now taking shape, there will need to be a need for special pharmacovigilance requirements for these new age medicines. And that is at a point where it is at a starting point, I would say. We are at an inflection point in the world of computing. This is where we are. There has been inevitable progress of technology. And there are two things that we must understand here. One is called speed and another is called friction. So what you see on the screen is the exponential growth of computing, and this is determined by a law called the Moore's law. So the world's maximum computing power doubles approximately every 2 years. That's the Moore's law. And then there is the friction. The compensator to the Moore's law is that successive generations of computer software, they increase in size and complexity. Thereby, they offset the performance gains predicted by the Moore's law. So this is the speed and friction, and that's how it plays. So, but the future is extremely, extremely exciting. There is quantum computing and there is DNA computing. What is quantum computing? So the basic unit of conventional computing is the bit. The more bits a computer has, the more calculations it can perform at once and the more powerful it is. With quantum computing, the basic unit of computation is called a quantum bit or qubit. So bits behave linearly. So for example, it will be to get a 20-bit computer, you add 2 plus 2 plus 2 10 times. But qubits are different. So qubit doubles the computing power. So a 10-qubit computer could actually be 2 multiplied by 2 multiplied by 2, which will make it 1024. That is the computing power. Now DNA computing is, so quantum computing is already a reality. DNA computing is the next thing that people are working on. And this is because DNA has its capacity to have extremely dense information storage capability. And everybody knows the structure of DNA, enormous parallelism, extraordinary energy efficiency. So if you have a computer built from DNA, then it can reproduce itself. It can run processes parallelly, and there is no problem of reinforcement. So if there's a problem that requires 1 billion calculations, rather than having one cell doing them all, you subdivide it into 1 billion cells working on one problem each. Imagine the power of computing that it would bring in. Let me give you another example of how the world of computing has and data analytics has changed and how it has influenced. The yo-yo test. Now, Virat Kohli, when he became the captain, the first thing that he introduced was a fitness test. And when they undergo a fitness test, what you notice on the screen here is wearing a black vest. Now, this black vest, it has an accelerometer, it has a gyroscope, it has a magnetometer, and it maps the player's movements in three-dimensional. It tracks directional change in the position on the field. And all this is fed into complex algorithms to measure the fatigue and training intensity. For example, what they did was the aerobic activities and anaerobic activities. So aerobic activities, they are activities where the body uses oxygen efficiently, and light or medium intensities like jogging or walking. But when there are fast activities like scrambling to get a run or catching a ball and all those things, they are anaerobic activities. So anaerobic activities run on fat stores and aerobic activities run on glycogen stores. And when they did all the analytics, they found that indeed fat stores are needed for anaerobic activities. That's how they changed the fitness level of the Indian cricket team. There are also, when you use technology, and let's say a computer is reading a case that has been an adverse event case that has been reported, a software is using. So the narrative says, I have 2 doses of drug X. How long does it take to start working? I've had no luck. The third shot is due on 21st August 2018, but someone just told me that it can't be administered until after Labor Day. Now the computer doesn't know that Labor Day is an important holiday in US, and this is what it meant, and it did not mean pregnancy. So context is extremely, extremely important. So that's the critical balance that is going to determine how efficiently are we able to use technology. A lot has been said about artificial intelligence, and this is the hype cycle for artificial intelligence. So there is the innovation trigger, there is the peak of inflated expectations, and then there is the trough of disillusionment, which is in many areas we have moved beyond this trough, and in many areas we are still in that valley where there is trough of disillusionment with that what it promised to do, will it actually do or not? And then you also, computers have to be continuously taught. They have to be given supervised learning. So what you see on the right is, will a computer be able to differentiate a puppy face from a blueberry muffin? So these are various things that will evolve over a period of time, but definitely the world is really exciting with all the various applications of AI that are already in use. And then there is another law. It's called the Amara's law. It says we tend to overestimate the effect of our technology in the short run and underestimate the effect in the long run. I think we are at a stage, we are exactly in the trough of disillusionment right now. And I think if we remain persistent, we will definitely enjoy the benefits of the technology in the longer run. And this is nothing to do with AI, by the way. It's just the visual representation of the enormous number of questions people usually come to me to ask when I talk big about the potential of AI. And one of the big questions that they ask is, how can we trust a machine that runs on a software to take such big decisions related to patient safety? And my answer to them is this. This is a view from a cockpit. How often have you all been flying in airplanes? Probably for decades now, many, many decades. Do you know that planes use auto landing system, and there is fog at the Delhi airport? Planes use CAT systems, computerized automated systems, which where the machine takes over, the software takes over, and you surrender your lives to the machine. My simple point is, if you can trust your lives with AI in aviation, I'm pretty sure over a period of time, you will begin to trust your data with AI and automation in areas like pharmacovigilance. Trust, but verify. That's what Ronald Reagan had said as well. Now, let's take a look at the patient of the future. Now, the patient of the future has a lot of variables now, and everybody, I'm very sure many of you, if we are 20 people in this room today, I'm sure 40 to 50% must be having some form of variable or must be aspiring to get some form of variable. So patients diagnosed with multiple sclerosis, the inputs from the variables, they go to the app, And then there are also wearable for tracking vitals. Brain scan is happening via wearable. Drones are delivering at least vaccines, if not medicine. There is, if a patient is enrolled in a clinical trial, then there is minimal disruption with the lifestyle, and she only has to visit the hospital for the important visits, only for monitoring of those aspects which cannot be done at home. So life is going to change. It is already changing. And then there are different kinds of wearable devices and connected devices. So there are the there's a broad classification, and these are like, let's say, in vitro devices, physiological monitors, mobile medical apps, wearables, and capital intensive devices. All of these are various things. I wouldn't go into the details of these, but what I want to tell you is that there is an increasing prevalence of connected devices in healthcare. And as you can see, the market is growing dramatically. It's growing by 32%. It's one, you must have heard of IoT, Internet of Things. This is Internet of Medical Things. That's booming right now. And with that, there are various examples. So for example, Otsuka and Proteus. So this is a partnership between a technology company and a pharmaceutical company. So they developed an ingestible sensor in a single tablet to record ingestion digitally, and which improves the drug compliance significantly. Novartis and Qualcomm, another example where the inhaler is connected to your app and it can monitor how often you have taken, compliance monitoring. Then at the same time, there is Dexcom and Verily, where they developed a continuous glucose monitoring device, and that can also transmit to an app. And therefore, connected devices connected to a software, making life easy for the patients, and a lot of these things can be monitored. So this is, I'll go into slightly more detail for this particular example. So smartphone compatible compact device, and accurately it's integrated with the app, and the data is then, there is real-time monitoring that happens in terms of the real-time alerts that can be given. Let's say there's a hypoglycemic alert, and it can send a family text message to the family with the GPS location and all included. So it may sound very, that is there a need to do? Yes, globally, this is happening. Of course, in a resource-constrained country like ours, we have a long way to begin to do such things. But yes, the world is changing. So this is my prophecy for pharmacovigilance in terms of the Internet of Things, the IoT of PV. There will be, for an outpatient, as you can see below, there will be, all these variables and all that post-authorization. Once a product is approved and you can monitor all that data, all these data, the parameters and all, they can be monitored. Data gets transmitted to a health cloud. And then a lot of these analytics, real-time advanced analytics can be done. Pattern recognition happens, an imminent adverse event is identified, and then an electronic health record is created, and then it goes back to the cloud, and wherever it needs to be reported, or whatever you need to do with that meaningful information, you proactively act on that information, and then you prevent an adverse event from happening if at all you can do that. So similarly, a thing can happen in an inpatient situation. All that data can actually go into a health cloud, where again, all these analytics can be run, and then you can use that data for in a meaningful manner. So let's see, how can one actually integrate IoT into pharmacovigilance? So there is data from a large number of users. Okay, basically more data results in more power to generate more evidence. And that's a very basic principle that all of us know. So how do these devices communicate to each other? So basically, the technology that makes IoT work are essentially machine-to-machine communication systems, such as wireless sensor networks like 2G, 3G, 4G, 5G, GSM, or the RFID, Wi-Fi. All these are various technologies where the communication happens between a machine-to-machine. And then there is NFC and RFID. The NFC is near field communication. You must have never used it on your phone, but it's mostly there on all the phones. And then there is radio frequency identification technology, which already exists. And this will increasingly, all these technology will become mainstream in our lives when a lot of this data will be communicated to each other. So incorporating advanced informatics is something which will happen. And there are the 3 V's of healthcare data. So 1 exabyte is actually equal to 10 raised to power 18 bytes. So this is a vast volume of data that is generated by all connected devices. And this data is characterized by 3 V's: volume, variety, and velocity. So volume is vast amount of data. Variety is a lot of heterogeneity in the data. And velocity is speed at which a data scientist can actually access and analyze all of this data. So advanced AI tools can actually be applied to analyze this data, and the tools that can be used are this machine learning, NLP, which is natural language processing, and various kinds of pattern recognition analysis that can be undertaken. So all these technologies are evolving right now, and all of these are going to shape the future. But there are problems. The problems are that the data needs to be first harmonized. There need to be global data standards that should be developed. And there needs to be data linkage. So there needs to be a linkage and mapping between relationship between the, let's say, the big data and the various other reporting systems. A common coding system needs to be developed. A lot of IT infrastructure needs to be invested in. And then at the end of the day, we need to have regulations which need to accept such data, and health authorities should be willing to accept such data. So a regulator of the future may look something like this, blockchain technology. If you're not familiar with blockchain technology, by the way, it's based on the same concept as a ladies' kitty party. There is a financial transaction that takes place. There is an element of trust built in the system, and the chain does not break. They continue the party. So a blockchain technology can be used by a regulator of the future in a way that when you send a large amount of data to a healthcare technology, let's say for a drug authorization, it can work in a manner that the transaction is secure and the transaction is fast. It's based on technology. It's not too much dependent upon a lot of resources, and that's how one can actually fast track applications in a manner that can be really amazing. At the end of the day, I would say one needs to be very careful by using all these technologies. Yes, all these technologies are technologies that will assist humans at the end of the day. And these are technologies that will not replace humans. They will always assist humans, because contextual knowledge is very, very important in all of these technologies. So as you can see on the left, here is somebody who's complaining to Indigo while their machine learning algorithm, their chatbot actually said, glad to hear that. And so this can really be very detrimental to somebody's business. And on the right, what you can see is how Amazon's machine learning technology and something which began to suggest more options, said that if you're going to buy a fan, might as well buy a noose as well to hang yourself. So one needs to be very careful with all of these. And that's exactly why this is a very exciting time when we can use AI and ML, machine learning, and all of these can be used to do a lot of analysis of data, but the human intervention can never be officially replaced in any sense of the word. So that's all I wanted to share today. Thank you very much.
Thank you very much. So the key message you are giving is a supervised data analytics and interpretation is the key for appropriate uses. Are there any questions? I don't see any question in the chat box. Maybe. Since there are no questions, I'll ask one question, and what is the benefits we are all talking about? Can you tell us risks which can occur in a pharmacovigilance context when we use AI or machine learning mechanisms?
Thank you for the question, sir. So the first thing, risk that is there is application of this technology without clearly understanding what does it take to apply this technology. So what we tend to do is we tend to create, it's like the planning needs to be good. For example, we get round pizzas in square boxes. It doesn't make sense because there may be a lot of wastage happening, and unnecessary the boxes are square in when actually we order round pizzas. So what we tend to do sometimes is that we first tend to create problems and then tend to solve those problems when actually they could be solved in the first place in the right manner. So linearity is a very important principle in application of artificial intelligence. So our process should be linear in nature. It shouldn't be circuitous in nature. It shouldn't be A to B, B to C, C to B, B to D, D to B, and B to A, and then A to E. It should be linear. A to B, B to C, C to D, D to E. The next principle one should apply is lean. So the process, do we need B and C also, or it can simply be A to D and D to E? So linearity and lean are two very important principles that one must look at before beginning to think on applying technology on any process. These are two things that we must do to our process, and then we should start thinking of how to apply this technology. The second thing is, we sometimes tend to believe that that this technology will actually replace humans. It is not going to replace us. So eventually it will only assist us, and the benefit should be done. For example, I'm currently doing a proof of concept where I'm comparing a particular process done by humans alone, and also going to do a time motion study to compare how will it be done by machines, humans assisted by machines. I'm going to not only look at the time that it takes, but also look at the precision and the recall values and the F-score to figure out that indeed the precision, accuracy, all of them are as good or better than humans alone. So, and then the quality of the data. The quality of the data also, one needs to assign those scores to be able to make that comparison. So one must plan well, one must see where it is applicable. Manual repetitive processes are areas where there is a higher likelihood of applying these technologies. So those are robotic process automation. And then there are machine learning and AI where you need to have contextual understanding as well. So one can a software read a paragraph and be able to decipher what is written in the paragraph by, and then you need to train that machine several times on that particular method, and that's called supervised learning, by which the machine begins to learn and repeats that same learning, what it has learned, and produces a better result every time. So all these are like the nuances of applying this process.
So there is one question that application and implementation and acceptance would clearly depend on the technology status of the country. So how, to what extent this is true, number one, and number two, what is the ethical dimension of using artificial intelligence and machine learning? Would you like to comment on these two things?
Sure, sir. So the regulators basically are beginning to accept certain things. There are various devices that they have already approved over a period of time, and where these artificial intelligence technologies are being used. So various medical devices have already been approved. So the regulators, in fact, the US FDA is laying a lot of focus on this, even more than what the EU regulator is doing at this point in time. And which is because usually it is the EU regulator which leads the world in terms of the pharmacovigilance regulation development. So there is a lot of acceptance, but the key question, as I had highlighted in my slides also, is trust. How will you build trust in the technology? And trust can only be built in the technology if more and more data is, it's like, you know, a comparator trial is undertaken between humans alone and human plus machines, and that data will build more and more trust and eventually show the world that yes, there is a lot of benefit, and that's how confidence will begin to develop, and more companies will begin to adapt to the situation and adopt these technologies. So there is always an adoption curve that is there whenever something new comes up. You remember the first time you started using a touch device instead of a key device alone. So there was an adoption curve that used to be there. Similar is going to be the phenomenon that is going to be working here. Now, in terms of ethical aspects, clearly ethics would play a role where, for example, if the data that is, how dependent is the data? How is the how dependable is the data? How can you really have the confidence that the data that's coming out can be used to be able to make big decisions like drug withdrawals, or maybe approving a drug sometimes using the data that is generated from the use of these technologies. So those will be the ethical dilemmas. And again, it goes back to the function of trust, where one will develop confidence in it and one will be able to take a call on that data.
Thank you very much. Thank you, Vivek. So over to Professor Usha Pingali. Usha ji, floor is yours.
Thank you, sir. Thank you, Dr. Arora. And it was very exciting listening to Vivek on this very interesting topic of artificial intelligence. I know there maybe have been more questions than, the answers. So we have the next speaker on benefit risk analysis session. He's none other than Dr. Viraj Rajadaksha. It's a pleasure always listening to you, Viraj. Viraj is going to speak to us on assessing benefit risks in real world with Candox 1 COVID-19 vaccine. Viraj is currently the Area Medical Director, AstraZeneca UK, where he oversees work in Middle East and Africa. He's an MD from Seth GS Medical College and KEMH Hospital. Viraj is a pharmaceutical physician and he has rich experience with several leading pharmaceutical companies, including Pfizer, Sanofi, just naming a couple of them. His areas of expertise includes women's health and diabetes, and Viraj hopes to develop innovative evidence generation capability programs, partnering with societies, health authorities, and patient groups, where he has a patient-centric approach to his program. And over to you, Viraj. I'm looking forward to hearing from you.
Good afternoon, and thank you, Madam, for those very kind introductions, remembering our frequent visits to Hyderabad as we were setting up clinical trials over a decade ago, and the real exemplary leadership of Dr. M.U.R. Naidu and you, both of you, in setting up clinical pharmacology as a separate discipline. Thank you, Dr. Nithya, for inviting me, and thank you, sir. Today, I'm going to talk about assessing the effectiveness and the possible risks. And this is more of a case study, an example that we have done over the last few months, I would say, or year. I will now share my screen. Let me see. Can you see my slides?
Yeah, Viraj, all good. I think you should just make it full screen. Yeah, go ahead, please. Please go ahead.
Okay, thank you. Thank you. So I'll be talking about assessing the benefit risk in the real world, and what are some of the lessons that we have learned over the last few years. Needless to say, I start with a disclaimer. I'm, of course, a full-time employee of AstraZeneca, and I was a part of the vaccine team at the area level when suddenly we were all possibly thrown under the bus and realized that we are facing one of the worst healthcare problems that the world has seen for nearly over a century. Of course, this session is purely for academic reasons. We must also understand that clinical practices as well as regulations can vary from time to time, even in a particular country. And my final thing is that while most of the opinions that have been expressed are based on existing evidence and are mine, they are not necessarily that of AstraZeneca. Over the last 24 months, the world faced possibly a catastrophe, the worst catastrophe since the Second World War, and probably the worst catastrophe since the Spanish flu hit us more than 100 years ago. And the way different partners in the world came together, first is to reduce the spread of coronavirus, though some measures were successful, some were not. And then later on the development of the vaccine is a standing example of what the world can do together when faced with a crisis. The vaccine was initially discovered by Oxford and later developed by our organization, of course, in partnership with various organizations, including Serum Institute of India in India. And the pivotal study, based on over 17,000 participants, was a very, very innovative phase 3 trial, because we had to develop the vaccine in less than 1 year, which ordinarily would have taken 2 to 3 years. But we had to come out with a solution, and we were not sure whether that solution would be the best possible solution. So this study was done in UK, Brazil, and South Africa with over 17,000 participants, and with those individuals followed up at least for 1 year. We showed that this study reduced significantly the incidence of COVID-19. On one hand, it provided protection against severe disease, hospitalizations, and death. And we must understand that when we want to control COVID from an overall healthcare system perspective, reduction in hospitalization and reducing the impact of healthcare systems was very important. In most of the Western countries, healthcare is a fundamental right. The state has to provide healthcare free of cost to individuals. And can you imagine if hospitals and institutions in the UK would have been flooded with thousands and possibly millions of patients, what would have been that situation? So a very clinically relevant endpoint was to find out whether it reduces severe disease hospitalization. We also found out that we needed 2 doses in most cases. In most of the vaccines needed 2 doses. And now we know that boostering of the vaccine is also necessary. But we also know that there was sufficient protection provided at least up to the time the second dose was given. And we also realized at some stage that if the interdose interval is about 12 weeks, then there was a slightly higher efficacy compared to the interdose interval, which is shorter. And there are many questions which sometimes are asked in the media, and there are questions which are asked to the government also, that why do you keep 12 weeks? You should vaccinate as many people as possible and you should give it in 4 weeks. But there was scientific data to suggest that if you keep the dose interval between 8 to 12 weeks, generally you do find better responses. And there were some exceptions made to it, but that was rather the rule. In another pivotal study in the US, we showed that with about 32,000 individuals who were included in this study, there was an absolute 100% efficacy against severe or critical disease and hospitalizations. And more important, we also got initial data that there is good amount of efficacy in 65 years and above, because that was a population which we wanted to protect. While randomized controlled clinical trials give us evidence, they still leave certain questions unanswered. And a very important clinical question that was unanswered in this analysis is, what is the effectiveness when it is given to particular groups of individuals? And the most important interested population here was population more than 60 to 70 years of age, and specifically more than 80 years of age. We must know that these populations are increasing in the West and countries like Japan. For the first time in Japan, we now know that the number of individuals who are more than 80 years of age are far more than the number of individuals who are less than 5 years of age, which means that the population has matured. And it is important for us to find out what is the effectiveness of this vaccine in these populations. So the vaccine efficacy overall, of course, increased to 82% when the interval was extended to at least 12 weeks. And this is not just an observation, but this is because when you understand how antibodies are generated and how the body's immune system responds to an administration of vaccine, you would realize that this interval of 12 weeks is clinically justified. Real-world evidence started immediately as the vaccines were available. And in the next few minutes, I will share some of these studies in more detail. And some of these initial studies done in the UK, because UK was one of the first countries where there was a massive rollout of the vaccine. In fact, within the first 6 months of 2021, nearly 80 to 90% of UK's adult population who were eligible for vaccines had received either the first dose, and by end of 21, nearly everyone had received the second dose as well. Public Health Scotland, which had a very well-developed system of collecting data, showed that in an enriched elderly population, especially those in care homes and institutions, there was a 94% efficacy in preventing hospitalizations. We also showed that in older adults, more than 70 years of age, it was not only hospitalizations, but also symptomatic COVID. And these are social questions because many of these individuals live alone and hence have, of course, we have to reduce hospitalizations, but if we are able to even reduce symptomatic COVID-19, it reduces the social impact of this disease on the broader population. And then another study which showed that the vaccine was 80% effective in elderly and frail real-world adults with extensive comorbid diseases. These were based on databases because we know that the NHS and the UK really have extensive databases which can be analyzable and reportable. Let's look at one of these studies, which was on Public Health England, on the effectiveness and impact of the vaccine. And this was done to estimate the real world effectiveness of two vaccines, the mRNA vaccine as well as the adenoviral vaccine, against confirmed COVID-19 hospitalizations and death. I will not go through the whole methodology in detail, but we looked at symptomatic PCR confirmed COVID infection, hospitalizations, and death, and it was a test negative case control design using logistic regression to estimate the odds of vaccination in confirmed cases compared to that of control. What this study showed is that one dose at that time of the adenoviral vaccine resulted in a reduction of cases as the vaccine was inducing immune activation. And this space of 12 weeks again was a very practical way of at least immunizing a far broader population with at least one dose, so that you have a broad population which at least has one dose, and then you start giving the second dose to obviously increase the effectiveness from 70% to probably 80-85%. And this is where science actually guided policy. And one of the messages that I want to give is, we do trials, we do real world evidence studies. This should impact policy. And in the UK, what we have seen is that the science that was generated, especially in the real world evidence, influenced how the government created the policy for vaccination. And this was probably then emulated across many other parts of the world. This study also showed that the effectiveness actually reaches close to 75% about 35 days after the first dose. And this is a very nice plot which talks about adjusted odds ratio. And as you start following up the patients for a long period of time, and of course, this is a 2021 publication. There are updated publications on this. You do see a far better effectiveness in the response. So it takes some time, but as you get into the second month of vaccination, you are really going to see visible effects of administration of this vaccine, which might not be that evident in the early stage. This also showed that the vaccine provided additional protection against hospitalization within 14 days of a positive test. You must understand that around early 21, Alpha variant was going down, while the Delta variant, which was initially isolated in India, but then spread all over the world, was actually spreading far more, and it was associated with far more complications. So we had to find out data. Is it really providing protection against hospitalization, and how fast can it provide protection? And this study showed that as early as 14 days after you administer the dose, you were really getting good amount of hospitalization reductions. And this helped the economy to come back to normalcy even after the administration of one dose. In Scotland and from England, hence, there was a very clear benefit that we saw in the reduction of hospitalizations. So finally, what did we see in this study? This study showed that vaccine effectiveness reached about 60% in the first month, but it was increasing to 73% in the second month. And it was increased. By the time you took the second dose, it was fairly high of about 75%. Secondly, it also showed the lower risk of emergency hospitalization, which was a very, very clinically relevant endpoint in that particular day. And this showed that real-world evidence was validating the results observed in the older population. And the number of older individuals who were there in the real world was far more than what you knew in the randomized controlled clinical trial. The next study that I will talk about is the effectiveness of the first dose of COVID-19 against hospital admissions. And just for interest of time, we will show that during the first half of 21, the uptake of both the adenoviral vaccine as well as the mRNA vaccine was pretty high across all older populations, especially nearly 100% in many of these older populations. And this study again showed that vaccine effectiveness increased over time, and it peaked at around, at the end of 1 month for both the vaccines with one dose. And then, of course, we know that this not only reduces the hospitalization, but when you give the second dose, that reduction of hospitalization nearly reaches up to 100%. So fairly good amount of protection in the first month. And when you add the second dose, this effectiveness is going to increase to about 100%. So overall, in the population which was more than 80 years of age, we could show that the combined vaccine effect in hospitalization with either vaccine, we are not here to discuss which of the vaccine is better, was very good. And that resulted really in a far reduction in the impact on the NHS system. And even during the worst days of the pandemic, the NHS as a system and many other systems in the world could cope up with the challenges that we are facing. So now we knew that the vaccine is working, not only in randomized trials, but it is also working in the real world. And you were really seeing visible results of the effect of mass vaccination that was happening. And then suddenly in March, we realized that there were some reports of thromboembolism, which especially came with adenoviral vaccines. So we looked back into our databases to see what are those results, and hence tried to do a very good assessment of effectiveness versus safety. Now we know that overall in the randomized controlled clinical trials, there were minor adverse events. It was generally a favorable safety profile. Early in the clinical development, there was there was a signal of transverse myelitis, which did not prove to be true. In fact, 2 of these cases were idiopathic. They were associated with spinal cord demyelination, but later on proven not to be associated with the vaccine in any way. So other than localized reactions which were there, the vaccines really did not produce significantly safety issues during the randomized controlled clinical trial program. But in the real world, the question then was, is there a higher risk of thrombosis with the vaccine? And the data that we collected was data from the yellow card system in the UK, where probably millions of individuals had received either of the vaccines. And this data showed that as of early March of 2021, and you might say, oh, this is March of 2021, but understand that millions of individuals have already received the vaccine in the first 3 to 4 months. We did see that there were very, very modest numbers of pulmonary embolism, infarction, and thrombosis. When you talk about thromboembolism, we need to focus on pulmonary embolism because it is catastrophic and it can cause mortality. Deep vein thrombosis occurs, but it need not always cause mortality. So this was a very, very clinically relevant endpoint. And we did show that there was not a significantly higher risk of pulmonary embolism. And similarly, there was also not a further risk in the overall thromboembolic related events, including thrombocytopenia, which could predispose an individual to a thromboembolic event. So overall, the UK spontaneous reports did not reveal any such increased risk of thromboembolism in the overall population. However, the question still remained that when you are talking about pharmacovigilance in a broader population, maybe it doesn't cause thromboembolism in a broader population, but there could be selected groups where this risk ratio might be different. And this was a question asked by the European authorities in March and April, and our teams worked very closely with them to try to find out and give a very scientifically validated answer. Is there a benefit of vaccinating a particular group versus the risk of thrombosis in the same group? And this is very important to answer, that you cannot look at benefit of vaccination in the older population and then look at risk of thrombosis in the broader population. This is like comparing apples and oranges. But we looked at specific work groups. And I have 3 or 4 slides which would look similar, but let me try to explain one slide and then the same principles apply to all. If you look at this slide, we are trying to look at the risk of COVID-19 hospitalizations, reduction probably, versus the prevalence or the incidence rather of unusual blood clots and low platelets. And the numbers on this part is cases of blood clots with low platelets. On the other hand, on the right side are cases of COVID-19 hospitalizations which are prevented. And we divide them into various groups. So obviously in the group which is 80 years and above, the risk of hospitalization was high. So when you administer a vaccine, the cases of hospitalizations that were prevented were very high. Let's say 1,239 per 100,000 population, while the risk of blood clots with low platelets was only 0.4. So you can see that the benefit to risk ratio, we always say risk to benefit ratio, we should also say benefit to risk ratio, was clearly in favor of administration of vaccine. When you look at slightly younger population, let's say 40, obviously the risk of hospitalization overall by COVID-19 is less. So when you give a vaccine, it reduces further, but the cases would be much lesser. Let's say it is 122, but it is still about more than 60 times better than the cases of blood clots that would remain. And even in the younger individuals who, because of social media experience, were saying that there is higher risk of thrombosis there, even them, the risk of giving, of having a thrombus is much lower compared to the benefit that you see with the vaccine. And this was seen in individuals who had a possibly high infection rate because of comorbidities, etc. But if you look at broader populations who probably have a low infection rate or also a medium infection rate, the benefits were always far more than the risks. And this is an interesting document that was submitted to the European authorities. You see the same effect on ICU admissions. In 80 and above, ICU admissions prevention was about 110, and the cases of blood clots remains as 0.4. So more than 100 times benefit seen with the vaccine. And finally, even in COVID-19 deaths, you can see that per 100,000 population, the vaccines prevented 733 deaths. On the other hand, the risk of thrombosis was only 0.4. So overall, across all the populations, there were very small populations in which possibly the risk of giving the vaccine was slightly numerically higher than what you see with the cases of blood clots. But overall, if you look at the broader population, there is a very clear benefit of hospitalizations, deaths, as well as severe COVID-19. So on the basis of that, as I come towards the end of my presentation, on the basis of this very well thought of study, the EMA as well as the UK MHRA reaffirmed the benefits of adenoviral vaccines. And they said that it continues to outweigh the risks because we were getting clear benefits on the reductions of hospitalizations. It was helping the economy to come back, and the risk of thromboembolism in the broader population as well as identified selected populations was far less compared to the effectiveness and the benefits that you were getting with mass vaccination. So I would like to end my presentation by saying that this is just an example of what an effective vaccine should be. And obviously the same thing will appear for mRNA vaccine as well. You should definitely have a vaccine which is safe. Most important thing, because you are giving it to individuals who are normal individuals, meaning they are not suffering from any disease necessarily. It should have very good amount of efficacy, but the clinical endpoints that you should look at could be very different in different stages of the pandemic. In today's scenario, you might still look at a vaccine which reduces symptomatic COVID-19 because that is the need of the hour. But early stages of the pandemic, you really wanted reduction of hospitalization. So our understanding of clinically relevant endpoints would also change. They should, of course, be good immunogenic. In a broader population, we know that vaccines may have a modest response in certain populations, but that's a topic for another day. And of course, the vaccine should be easily administered and able to get into low and middle income countries as well. So finally, we have to be vigilant. There is no question about it. And you have to be prepared to walk, but be ready to stop. And these questions will come over. There were some controversies related to myocarditis in mRNA vaccines, and to some extent, you then know what are the groups of individuals where you have to be careful. Does that mean that you need to do a mass-based assessment for every individual? No. As physicians, as policymakers, we need to understand that the benefits of giving the vaccine in a broader population are far, far more than the anticipated risks. And if there are individual risks with individual patient or an individual, it is important for us to have a dialogue and then take an informed decision whether to proceed or not to proceed. I thank the chairperson, sir and madam, for giving me this opportunity to talk. I know this is not the end of the journey for COVID-19, but we will fight this journey together, all of us in collaboration, and look forward to meeting each of you personally in one of my next visits. So thank you so much. And I will request if there are any questions, I would like to address those questions now.
Thank you, Viraj, and thank you for joining, at this early hour. I know, I'm sure it must be around 7 o'clock in the morning for you on a Saturday, but thank you very much for joining us and giving your personal experience with this molecule. And it was really exciting, and it was an excellent presentation. And yeah, we really understand that there were a lot of challenges in this whole program because it was something very different and something very new. And I would just like to ask you, like, since there are no questions specifically in the chat box, what were the challenges you people had, especially, when dealing with elderly patients, because they will be having a lot of other comorbidities. So specifically when you're, especially in relation to the safety profile, were there any challenges in safety reporting, especially in the elderly receiving?
So luckily what happens is in countries like UK, obviously, you have the yellow card system, and the way reporting happens, it is far more robust and far more reliable. In fact, what we saw is that the vaccine uptake in the elderly individuals was very high. And this was because they could anticipate the risk of hospitalization, and they had seen some of their inmates or some of their own friends, suffering from that. So in fact, from the elderly, even within the early few days, we saw a very good reporting systems, because either it was reported through the social care system where they were living, or they were in regular touch with their GPs. But what is also important, madam, is that we need to see similar reporting systems all across the world. Because the data that I presented to you was coming from mainly European and UK centers. And India is probably one of the countries where the number of individuals vaccinated would be far more than the number of vaccinations in whole of continental Europe. And it would be really useful for us to understand that, is it the same scenario? It could be. Or is the scenario slightly different? And in India, probably that would be a challenge that we should add. But if we are able to get that data, I think it would be one of the most robust data sets, far more, in terms of quantity compared to what I've shared.
Yeah, true. Yeah, we agree.
Yes,
sir. Over to you, sir.
So thank you, Viraj, for this presentation. And you are aware that India is one of the few countries where interval was kept between 12 to 16 weeks. And there was a lot of debate and a lot of pressure that it should be reduced, but we maintained. And in the long run, we think that was a wise decision, in the sense that there is a vaccine tracker platform for the country. And it shows probably the delayed second dose was giving a good protection. And today, whatever data as of January, that even if a person has received single dose, protection against death is 97%. And if 2 doses are given, protection against death are 98.7% or something. So the vaccine, and you are aware that 90% of the vaccine used, or I think cumulatively, globally, overall AstraZeneca maximum doses have been given in this country. TTS has been seen. I'm sure you are aware of those data. But our pharmacovigilance or AE5 surveillance is able to capture data for the first week only, not beyond that. While a lot of cases would have occurred between 4 days to 21 days or 24 days, whatever that interval is. But we have seen, but the number of cases were less. In fact, there were a couple of cases with even Covaxin also. So, but I think Covishield has done a lot of good to this country. And the whole question is, why, despite such large usage, and probably UK and India are the biggest consumer of this vaccine globally, your company is having difficulty in selling it to the new countries. In COVAX facility, a lot of people are refusing to take this vaccine. What is the reason for that?
Yes, so again, as I said, I'm speaking today personally, and in my
yes, so I do understand that.
Yes,
but what is
there were, of course, several issues, and even if you look at low income and medium income countries over the whole of 2021, we supplied nearly 70% of COVAX supplies actually came from either the direct AstraZeneca vaccine or through Covishield, through our partners Serum Institute of India or R-Pharm in Russia. So throughout 21, that has definitely happened. Now, as we were getting into the Omicron variant later, there was a need definitely of giving the third booster. And there was some data to suggest that possibly heterologous boosting, could be very useful. Of course, our uptake in Europe was quite less, and I must accept that our vaccine does not exist in the US. It has been submitted, but we have not got a decision on the US. So as a result of that, obviously, as we started getting into the booster phase, there was an acceptance of other vaccines as well, because the base vaccine which individuals had received was the AstraZeneca or the SII vaccine. So then if you have to give an heterologous boosting, then you need to give a different vaccine as well. And so the third dose came predominantly from either the Pfizer or the Moderna vaccines. Irrespective of that, it is important for us to see that people have to be vaccinated. Which vaccine is not the most important thing, but they have to be vaccinated, and at sufficient time, they have to also receive the boosters. But yes, there were certain challenges, not necessarily all of them were scientific challenges, but there were challenges in certain geographies, not in the emerging parts of the world, but predominantly Europe and US.
Yeah, so basically, I also wanted to say, because there is this perception about vaccine for poor and vaccine for rich, and which is in fact absolutely wrong. But I'm still concerned that in Africa, for example, only 15-20% have got their primary schedule. 80% are still there. And a vaccine like Covishield or AstraZeneca is more aligned with the ground realities. But still people want to have Moderna and Pfizer, which cannot be given beyond even capital cities of most of these countries. Forget about other urban areas. So
I agree with you, sir.
Is there a marketing failure on this part of AstraZeneca?
No, I agree with you, sir. And I think when you look at some of the benefits of the adenoviral vaccines, especially related to storage and administration, this actually seems like a vaccine which solves the problems of the developing world. The issues in Africa were not only related to the supply, but also the whole distribution system. Because in many parts of Africa, there was, of course, as you rightly said, the vaccine, because of social media influence. And at the end of it, there were vaccines available, but the uptake of those vaccines at some stage was very modest because of misinformation. And as a result of that, you do see isolated pockets of high prevalence of COVID infection coming from Africa, or the way some of the other countries have returned back to normalcy, you really do not see that. So that is really a lesson for us. We were all new in this journey, and probably this is a lesson for us going forward.
Thank you.
Can I ask a question, Dr. Arora?
Please, Nilima ji, namaskar. Please go ahead.
Namaskar. Very wonderful to see you, Dr. Arora and Usharani. Thank you, Vivek, for that very excellent presentation. I have just one question. The WHO webinars have been recently, in the last couple of weeks, have got scientists from all over the globe working onto the effects of vaccines versus various strains like Delta and Omicron and the first strain and so on. And the data collected in terms of the levels of antibodies, or in terms of the T-cell response, or in terms of even the animal studies, indicate that these vaccines are all effective even against Omicron. When we look at the T-cell responses and what they have through these in vitro studies. However, and you mentioned that the drug, this AstraZeneca vaccine, is 100% effective, and even in the effectiveness study, it has nearly 94-95% effect in reducing the hospitalization and death. But in India, we find that we do have reports of deaths occurring, and of course, I have to admit that what we have are like anecdotal, when we have our own colleagues and doctors and family members who have succumbed to COVID despite 2 vaccines or sometimes even 3 vaccines. I mean, the third dose as well. So I don't know how you can put all this data together, and what is your really comment on that? I would really like to have your comment on that. Thank you, sir.
Thank you, madam. It is really a pleasure to see you, and we have all learned from you at all stages of our residency life and later. This is a very important question, madam, about, yes, the data that I shared with you was on randomized controlled clinical trials and effective and real world evidence studies. Predominantly, of course, the most prevalent variant at that time when this data was generated was initially alpha and then delta. During the randomized trials, it was alpha, and then it was delta mainly during the real world evidence studies. With Omicron, there is good amount of data from in vitro studies that there is very high effectiveness of the vaccine. However, there is also an interesting paper that has come out that there are certain groups of individuals in whom the effectiveness of the vaccine is far modest than the general population. And these are individuals with chronic kidney disease, either transplantation, patients undergoing chemotherapy with past hemato-oncological malignancies. And there is also a small population with chronic obstructive pulmonary disease and cardiovascular disease who might have a slightly modest response to some of the vaccines. Now, while this number of population has been estimated to be 1.5% of the overall population, but when you multiply it with 1.2 billion people, this becomes a substantial number. And hence, and now I'm not speaking from an epidemiological perspective, but I'm speaking from a clinical perspective, that when you look at individuals there, and if you feel that you have an individual who has that higher risk of development of COVID-19 hospitalization, or the vaccine effectiveness itself could be modest, are there other ways of providing protection? Just another approach could be monoclonal antibodies. It is not cost effective at a population level at all. But for individuals, there might be a higher protection that could be given by these other group of medications. So this could be possibly, my answer to this. Of course, we must also be vigilant that as the new variants arise, what is the effectiveness of these vaccines? For example, we know that with the beta variant in South Africa, the effectiveness of the vaccines was far more modest compared to what you see with alpha and delta. So we should be vigilant about future variants as well.
Dr. Arora, may I make one more point here, please?
Please go ahead. Yeah.
There's also something which is called as the onslaught of infection, or the force of infection, as some immunologists put, force of infection. So, so we often find that healthcare workers who probably have more force of infection because they get exposed to the environment despite all the PPE and everything, whether the mortality is more in these individuals. I will just digress for 1 minute to give the study which ICMR has done with all our centers across the country. We had the study on 44 centers with 12,000 individuals. Now, this was at the beginning of the COVID pandemic, when ICMR had put in its advisory hydroxychloroquine as a pre-exposure prophylaxis when vaccines were not available. And we did this study so as to be able to support, or whatever, the ICMR guideline of hydroxychloroquine being used pre-exposure prophylaxis. So we had 12,000 healthcare workers, of which about 1/3 were actually taking hydroxychloroquine and 2/3 were not taking. And this is just as as you said, real life data. And one of the observations that we found was when we, did the regression analysis and so on, that we had the spread all over in India. So we noted that in a place like Bhopal, where the infection rate was not very high, the positivity rate of those tested was not very high, the effect of hydroxychloroquine was, quite substantial. While in a place like Mumbai, where the positivity rate was very high, the effect was less. And also similarly, patients who had comorbidities, as you also correctly suggested. In them, the hydroxychloroquine effect was less compared to those who did not have the comorbidity. So I wonder whether this, you had the randomized control study, but they were done in 3 countries. So, and the positivity rate at given time may be different at different countries and at different time points. Whether this was also an observation that you had, and whether this has been specifically looked at, because this was our very definite, observation in that hydroxychloroquine study that we did.
Yes, absolutely, madam. And I think while there is no clear data on that, but yes, it's an important scientific hypothesis. And it would be important because we do have data from these individuals. And over the next few years, I think we will understand this pandemic much more. And what should be our reactions in the future and how do we prevent it. So while currently we may not have that data offhand, but this will be a very important point. And I will reach out to you separately just to understand, what data sets we can use for. But yeah, I agree.
Thank you. So we have just made a short strategy article in the JAPI Journal of Association of Physicians of India, looking at all these aspects when deciding on to the pre-exposure prophylaxis. But thank you. Great to hear from you, Viraj, and a wonderful presentation. And thank you, Dr. Arora, for giving me this opportunity to ask this question. And thank you, Usha.
Thank you, ma'am. It's a pleasure. And since we're running short of time, thank you, Viraj, for the wonderful presentation once again. And thanks, Nithya, for giving me this opportunity. And over to you, sir.
Thank you very much, Viraj. And it's such a pleasure hearing you. We have the last speaker of the session, Dr. Robin Ferner, who is Clinical Pharmacology, City Hospital, Birmingham. The important point is that he's a close friend and associate of Department of Clinical Pharmacology at KEMH and has been teaching there for a long time. So it's really an honor to welcome you, sir. And you have a special interest in areas of medication errors. And today we are going to hear from you benefit and risk assessment of mefloquine, a UK perspective. Over to you, Robin. Thank you very much.
Well, thank you very much. And it's a great pleasure to be in Mumbai, even if I'm only there virtually. I'm going to try and share my screen. Can you see the slides now?
Not yet, no.
Oh. How's that?
Yes, better. You just need to put it on the full screen mode, I think,
And
yeah.
So we're nearly there. And as I say, it's a pleasure, and it's also been a pleasure to hear the last 3 presentations. I hope you'll forgive me that I didn't get up any earlier than that, but the last 3 presentations were stunning. As background, I have to say that cost-benefit analysis is very difficult. It needs good estimates of the numerators, how many cases of harm there are, how many cases of benefit, and of the denominators, how many times, for example, a drug has been prescribed, but we don't have either of those for mefloquine. And so much of what I'm going to say will be qualitative. That's partly reflected in the talk we've just heard about COVID and the different variants of the SARS-CoV-2 virus, because that has meant that the benefits of the vaccines have changed with time. Not only that, AstraZeneca was the first vaccine available here in England, but now there are several others, and so the relative benefits, as with mefloquine, have changed with time. So it's a useful introduction, I might say. As part of the background, it's worth saying that malaria is very rare in North America, Western Europe, and Japan, though we do occasionally see it imported, whereas pharma is mainly in those places. So pharma has been little interested in malaria. And much of the early development has depended on other things. This is a map showing how things were in 2018. And they've changed dramatically, even since I first went to India, and Nilima Kshirssagar, Madam, arranged for me to go to Kaustubha Hospital in Mumbai, where there was a lot of malaria. I gather that now in India as a whole, malaria is rarer than it was 20 years ago. Much of the history of anti-malarials depends on conquest and war. So it was the Spanish conquistadors in Peru who first exploited quinine. In the Second World War, proguanil and chloroquine for war in the Far East, and in the Vietnam War, the drug that we're going to talk more about, mefloquine. One exception, which is now very important, is used exploitation of a traditional Chinese medicine to elaborate artemisinin. So here's the structure of mefloquine, and just to point out that there's an asymmetric carbon atom, as there is in quinine, and that there are, as a marker of fat solubility, if you like, 2 trifluoromethyl groups. I'm not going to go into the life cycle of the malaria parasite, except to say that mefloquine attacks the intraerythrocytic stage and prevents the development of schizons. The precise mechanism is not yet known. Absorption is good. The distribution volume, as you'd expect, is high, about 20 liters per kilo. And post-mortem concentrations show that it's mainly in the liver. They also show something important for the rest of our discussion, namely that in the brain there is a substantial amounts of mefloquine, at least post-mortem. And further experiments in rodents demonstrated that after repeated oral dosing in rats, the amount of mefloquine in the brain was high, and it was particularly one optical isomer rather than another. Barreau de la Gagerie extended these experiments with intraperitoneal injection in mice, and she found that firstly, there was a lot of mefloquine in the brain. This is the brain-blood concentration ratio against time. And you can see that at the peak, there is about 4 times as much mefloquine in the brain as there is in the blood. And second, that, as had been found before, it's mainly the minus isomer and not the plus isomer. What she also demonstrated was that if you block the efflux transporter P-glycoprotein, the multi-drug resistance transporter, then you get much higher concentrations in the brain still. In other words, that the blood-brain barrier has to be intact to keep mefloquine concentrations in the brain low. And in humans, Ruvex had 5 patients with cerebral malaria, which of course might affect the blood-brain barrier, and they had an acute brain syndrome after treatment with mefloquine. They were able to measure mefloquine in the CSF in one patient in very high concentration. In another patient, they couldn't find mefloquine, but they did find the metabolite, and they concluded that mefloquine can cross the blood-brain barrier, and that's in man. Metabolism is hepatic, and particularly cytochrome P450-3A4, but others as well, to produce the major metabolite mefloquine carboxylic acid. It has a very long half-life of about 3 weeks, but this is uncertain and it's definitely variable. 3 studies show a range from at the least 6.5 days to at the most 33 days in different volunteers. And it seems to be shorter in Thai people than in Swiss people, perhaps because the Swiss have more body fat. As with other drugs, there have been different phases of investigation, and the sinister story behind mefloquine is that having been developed by the Walter Reed Army Hospital in the States, the first studies in man were carried out on prisoners at the Statesville prison in Illinois. I was going to say Victorian, but at any rate, a 19th century institution where people were essentially locked up in cages. And perhaps it's too much to describe them as volunteer studies, but the investigators gave 19 doses from 5 to 2,000 milligrams to patients, each dose 2 active doses and 2 placebo doses, and they compared effects and measured pharmacokinetics. And they reported at 1,750 and 2,000 milligrams, all 4 subjects who received the drug reported transient dizziness and nausea within the first 48 hours. So this didn't deter them from giving experimental treatment to patients whom they actually inoculated with Plasmodium falciparum of different strains, and found that whilst the highest dose effected a cure, lower doses did not. They don't mention adverse drug reactions at all in this experiment, and nor do they mention how they dealt with those patients who weren't cured by mefloquine. This, I have to say, was published in the journal Science, which is one of the most important scientific journals in the United States. So it shows how things have changed. There were field trials, mainly in Thailand, and then the reports were thin, I think it's fair to say, before a prophylactic trial in Thai subjects, nearly 500, and they were well protected compared with the 36 subjects who were given placebo, and all of whom had at least 1 attack of malaria, sometimes several. The drugs were well tolerated, with no instances, said the authors, of untoward clinical reactions. Another study, also in Thailand, showed that the higher the dose, the more likely you are to avoid relapse. They did report 1 adverse effect, but it was late. It was 21 days after receiving the highest dose of mefloquine. Now, you will already have deduced from the fact that the half-life is around 21 days that there was still a substantial amount of mefloquine in the system, but whether this was due to the drug is a matter of debate. A treatment trial in Zambia showed that actually back in 1983, mefloquine in red was slightly less effective than chloroquine, which is surprising to our ears when one talks of falciparum malaria today. They did observe substantial adverse effects. 22% of the patients had dizziness and 1 developed behavioral disturbance. With chloroquine, a high proportion developed itching and abdominal pain. The Dutch looked at non-immune travelers given either mefloquine or atovaquone proguanil, let's say Malarone, in quite large numbers, though not all of them took the treatment and fewer still completed treatment, and they found more in the way of treatment-limiting adverse effects with mefloquine, most of those effects being neuropsychiatric. So this was some confirmation of the fact that neuropsychiatric adverse effects can be quite important in patients treated with mefloquine. Young Karen patients given higher doses for treatment developed more adverse effects at a later stage than those given lower doses. So perhaps unsurprisingly, the adverse effects were worse at higher doses, though the treatment failure was higher at lower doses, showing, I think, at least in these patients, how there's a trade-off between the costs of adverse effects and the benefits of treatment. None of this really worried people in the Western world until Pachen in 1989 gave treatment doses of mefloquine to 7 volunteers, and all the volunteers in the States developed lightheadedness or difficulty in concentrating or nausea or dizziness or vertigo or all of these. And the symptoms lasted for up to a fortnight. We found an unexpectedly high rate of adverse drug reactions to treatment doses. And after this, a survey in Germany found rather similar problems, both with prophylaxis and with treatment for non-cerebral malaria. This study was important because the number of patients treated for malaria in Germany was known, and the symptoms of psychosis, hyperactivity, hallucination, seizure, and disequilibrium were themselves significant. They occurred some time after the therapeutic doses. They lasted for several days, and the incidence could be calculated as approximately 1 in 215 of patients treated in Germany. So about 0.5%. An interview survey in travelers from Switzerland showed about 8% had neuropsychiatric adverse effects. Hello?
Hello. Dwella, please mute yourself. Sorry, Dr. Fun. Please go ahead.
Not at all. No, I was worried that someone was trying to tell me something. And you can see that most of these adverse effects occurred with the first or second dose. Treatment doses in healthy volunteers, which you remember Pachen had first described, again, was studied by Randy Wagner, and again, there were very high rates of adverse effects, which lasted much longer in women than in men. So the efficacy of mefloquine, doxycycline, atovaquone, preguanil, and other antimalarials was examined by Tickle Painter in a systematic review in the context of prophylaxis. And what this group showed was that all these drugs appeared to be highly effective, though the studies they looked at were relatively old, and it's now clear that resistance to mefloquine, particularly in some parts of Southeast Asia, is important. So that was efficacy. What about safety? Well, when comparing mefloquine with malarone, there were, as you can see, significantly higher risks of the neuropsychiatric adverse effects: abnormal dreams, insomnia, anxiety, and depressed mood. Now, that is particularly important if you're a short-term traveler from Western Europe going to a malarial zone, and possibly for soldiers trying to fight in malarial zones who are not used to these conditions. A second systematic review by Bitter noted that mefloquine affected more domains of mental and neurological manifestations than any other antimalarial drug, although they came up with rather different values for different drugs. So 4% of patients given mefloquine developed psychiatric or behavioral difficulties. 15 had mild neurological disturbance, which include, I think, vertigo and sleep disturbance in 1 and 8. Personality change, whatever that may be, is rare. Headache is common. Visual problems, which are especially significant for airline pilots, have meant that the aviation authorities have banned mefloquine from flying crew, in particular from pilots. So in avoiding adverse drug reactions, such as the neuropsychiatric adverse effects of mefloquine, one question is, can you identify those people who are susceptible? And our mnemonic for susceptibility, gasped, genetic, age, sex, physiology, exogenous factors, and disease is relevant here. I've already explained that the blood-brain barrier is important. And there are differences in the genes that code for P-glycoprotein ABC1B, also known as MDR, both in the malarial mosquito and in humans. So firstly, mefloquine resistance in falciparum malaria, in Plasmodium falciparum, is probably mediated by MDR. And second, humans with relatively inactive P-glycoprotein, for genetic reasons, may be especially susceptible. Metabolism may also be relevant. With regard to age, young people seem less susceptible than older people to the neuropsychiatric adverse effects of mefloquine, and that may be because they're protected in some way, or it may be that they are less fearful of developing neuropsychiatric adverse reactions, which is one of the problems that bedevils this particular study, that anticipation of neuropsychiatric adverse effects may cause neuropsychiatric adverse effects. What is clear is that, the reasons are uncertain, but what is clear is that women are more susceptible to the adverse effects than men, and they're longer lasting in women than men. It may simply be a question of dosage, because women on the whole weigh less than men, but that's not certain. Physiological factors haven't been identified, but it looks as if mefloquine is safe in pregnancy, which is encouraging and helpful. There is some suggestion that if you take mefloquine with alcohol, then that increases the likelihood of neuropsychiatric adverse effects. And since soldiers, when off duty, drink heavily, this may explain some of the difficulties that soldiers have had. There is a question as to whether pre-existing psychiatric disorders make you more prone to develop neuropsychiatric adverse effects of mefloquine. Whether that is so is not clear, but the safety data sheets for these drugs, for mefloquine, say that this should be an exclusion criterion. So other important adverse reactions, which probably don't weigh heavily in the balance because they occur with all drugs, all the relevant drugs, Stevens-Johnson syndrome, toxic epidermal necrolysis, liver injury, which seems to be commoner with malorone than with mefloquine, and arrhythmia, which is usually benign and which is self-limiting. So they don't weigh heavily in the balance. Where does this leave us? Well, we know malaria can kill. We know that prophylaxis cuts cases, though bite prevention with bed nets, proper clothing, and DEET is probably the first line and drugs are second line. While earlier in its career, it was said that from the pharmacokinetic point of view, mefloquine is an ideal medicament. The fact that it lasts a long time means that it gives long-lasting protection, but also long-lasting adverse effects. And if the adverse effects last for a fortnight, and that's the duration of your holiday, this doesn't make it an ideal drug for travelers from Western Europe to zones where there's malaria. I think the evidence is quite strong that there are rare but serious psychiatric adverse effects, but I've tried to hint at some of the difficulties of distinguishing anticipation from real adverse effects. And there is now increasing mefloquine resistance. So that's prophylaxis. With regard to treatment, mefloquine has generally been superseded by artemisinin-based regimens. My conclusions, firstly, the very variable kinetics and long half-life is a two-edged sword. It does get to the brain, it's not universally effective, it commonly causes nightmares. and it rarely causes serious adverse psychiatric effects. There are alternatives, doxycycline and malorone. Both have common but minor adverse effects. Doxycycline causes photosensitivity, which is particularly a difficulty if you're talking about Western Europeans going on holidays where they wish to get suntanned. Both drugs have to be taken every day, and malorone is costly. So I'm going to end with the conclusion that Nithya Gupta came to in her BMJ leader on the use of mefloquine in armed forces. The US Army scheme seems logical. If tolerated, use doxycycline, which is cheap and effective, although you have to take it daily. If not, etovaquone proguanil, which is effective but expensive, and again, you have to take it daily. And as third line, mefloquine, which you have to take weekly, but which is sometimes ineffective and which rarely causes adverse psychiatric and neurological adverse effects, which are a bad thing if you're in the army, and probably anyway. So thank you very much for letting me have this brief tour of the costs and benefits of mefloquine. They're still not absolutely clear. If it's of interest, if and when I need malaria prophylaxis, I usually take doxycycline. Thank you.
Thank you very much, Dr. Ferner, and we thank you for joining us at this early hour. I know it must be pretty early in the morning. You've joined all the way from UK. Thanks a lot for that. And the presentation was really excellent, and you had very nicely summarized the neuropsychiatric ADRs of mefloquine with your GASP factors. Very nicely put that across, and appreciate the presentation. And then one thing which was interesting is that women had more adverse effects compared to men. So that was a very interesting aspect. Is it because of any adverse drug reactions, or I'm sorry, is it because of any genetic factors, or?
This is a really interesting question, actually. And it's been raised in the context of other drugs as well. And so there are several possibilities. One is that women are more likely to report, for example, to the yellow card scheme than men are, because they're more fastidious, they're more interested in health matters, and they believe that the public good is served by reporting adverse drug reactions. That's possible. So the reporting rate may be higher. It is also possible that women are less tolerant of adverse effects than men. So whilst men, for example, if they feel a bit dizzy, just carry on, it would be possible that women are more likely to report that as an adverse effect. But I think neither of those is a good explanation, actually. I think much more likely is that at the doses given, there are physiological or genetic reasons for women to suffer more harm. Now, if it were true that it were entirely due to dose, and if you had good data, you'd expect that women would also benefit more, because both the benefits and the harms are related to dose. But I don't believe, for example, with antihypertensive drugs, that there is good evidence that they work better in women than in men. Perhaps others in the audience will know.
Yeah. Thank you very much, Dr. Ferner. And I don't see any questions in the chat box. So over to you, Nithya.
Madam Shirsagar is there. Do you have any comment? Yeah.
And no, only one observation that some beside the neuropsychiatric, I don't know, neuropsychiatric seem to include more the psychiatric, but there are reports of neurological side effects as well, which could be of the nature of neuropathy. And these sometimes have been reported to last a long time, especially if taken for prophylaxis. And such patients often undergo a whole lot of gamut of investigations, including MRI and whatever. And so that's another dimension of the ADR to mefloquine, I thought. And forever we are indebted to Dr. Robin Pernow for all his wonderful support to the department. And Nithya would definitely make more mention of that. Thank you, Robin, again.
Well, thank you, Nilma. And yes, I think it's a moot point, for example, whether vertigo is neurological or psychiatric, but what you say is obviously true. And the early reports with treatment of seizures are a bit difficult to distinguish between the effects of cerebral malaria and the effects of the drug. But it's reasonably clear that mefloquine can cause seizures in a small number of cases.
Thank you. Thank you.
So thank you for reminding me of these important facts, as ever.
Shruti?
Thank you, everyone. I thank the speakers of this session, Dr. Vivek Ahuja, Dr. Viraj, and Dr. Ravid Farnan for the wonderful session, and also the chairpersons, Dr. N.K. Arora and Dr. Usha Rani Madam for moderating the session. Now I would like to propose the vote of thanks. Good afternoon all, and it gives me great pleasure to propose the vote of thanks for this advanced level training workshop in pharmacovigilance based on the art and science of benefit risk analysis. At the outset, my sincere thanks are due to Dr. Sangeeta Rawat Madam, our Dean, and Dr. Shenvi Madam, our Academic Dean, for their kind support for the workshop and their valuable inputs, which gave shape to the workshop. Next, our thanks are due to Dr. Rajiv Raghuvanshi, who is Secretary cum Scientific Director of IPC Ghaziabad, for consenting to speak and chair a session at this workshop. We are grateful to Sir and his team, especially Dr. Jayaprakash, for all the help and support rendered towards the organization for the meeting. This workshop was held under the aegis of the PVPI. Pharmacovigilance is only possible with the support of our clinicians, and we thank all of them who have contributed to our AMC for over the last several decades, especially from the departments of medicine, pediatrics, hematology, endocrinology, gastroenterology, pediatric surgery, and neonatology. My sincere thanks are due to all speakers and chairpersons for their contributions, which led to great learning experience over the last 4 hours and dissemination and understanding of the complex area of benefit risk assessment in pharmacovigilance. Thank you so much, everyone. I am also grateful to Dr. Geeta Madam, who is Professor and Head of Microbiology, and her team from KEM for the use of the Resource Center facility, which made the UT team, Mr. Badal and Mr. Tushar, for this. I also extend my gratitude to Dr. Nithya Madam, who is Professor and Head of Clinical Pharmacology Department, the Pharmacovigilance team, and my colleagues for organizing such workshop. Last but not the least, India. Without you, this learned from the resource persons. With this, I call the workshop to a close. Thank you one and all. Stay safe, stay well. Bye everyone. We can log off.
Thank you, everybody.
Thank you.
Thank you, madam. Thank you, Dr. Farna, Viraj. Thank you to all those who are still online. Thank you very much. Bye-bye.
Thank you. Bye.
Thank you, Usha. Bye.
Bye, everyone.